Good morning or good afternoon, depending where you are. We have a busy schedule, so we'll jump right in. Next. So welcome to SDS POPs 2025. This is an advocacy and partnering summit. So our goal is to bring patients, researchers, and clinicians together to discuss all things related to SDS. Next. So this is hosted by us, the Shwachman-Diamond Syndrome Alliance, which is a US-based nonprofit serving the global SDS community to improve and save lives of people affected by SDS by focusing on research and therapy development. Next. So as you all know, SDS is a serious life-threatening condition. So this is the problem we are seeking to address. The problem we are most concerned with at this stage is the risk of developing leukemia. It's estimated to hit about 30% of patients by age 30 and more after. More recent estimates say 50% by age 50. Next. So our mission as the SDS Alliance is to give SDS families more birthdays to celebrate. We want to save lives by accelerating therapy development. Next. So as mentioned, this is SDS POPs. This is our annual virtual global patient summit. And the patients are really at the center of everything we do. Next. So I'm your host or MC today. I'm the mother of a child with SDS. She's 11 years old now. So this picture is from a few years ago. I'm also a scientist, a molecular biologist, and I happen to have done my Ph.D. actually on acute myeloid leukemia and the mechanism with which it develops. And 5 years ago, I founded the SDS Alliance for the purposes I just mentioned. Next. So also with us today is Jeff Cooper on the tech side. Many of you may have met him at our PFDD meeting back in June in Cincinnati. So he is behind the scenes helping run this meeting smoothly. If you have any technical issues, you can type them in the chat. Next. Now to the housekeeping items. So we are using Zoom today, as you all know. Many of you are familiar with it, but just wanted to point you to the controls and tools. They will appear at the bottom. For the main portion of the presentations, the audience will not have the ability to unmute themselves or show themselves on camera, but the speakers will. So if you have any questions or comments, you can type them into the chat box. That will also apply for when we have Q&A sessions. We will look at your questions and read them out loud and discuss them. Captions are available and translations of the captions is also enabled. I'll show that in the next slide. We are recording this meeting, and that will also help with watching it in the future in other languages. But we will remove all patient identifiers. Patient privacy is, we take that very seriously. Please don't offer any medical advice, and please adhere to our community guidelines. They're linked in the footer of our website. And after the meeting, we will send you a survey to provide feedback about SDS POPs so that in the future we can keep on making it as impactful for everybody as possible. Next. Next slide. So how to use the translations of the captions. So first you have to turn on the closed captioning. That's the CC button on the bottom. And then you should be able to select, there should be an up arrow and further options to then select what language you would like the closed captions to be translated into. So the speaking language is English for today's meeting, but you can choose in what language those captions should be translated to. Also, a quick shout out to our partners and sponsors. Of course, your contributions were key to making all this work possible, but we also had very impactful funding from the Chan Zuckerberg Initiative, from PCORI, and some research partners and sponsors as well. So if you have connections and your network is able to possibly step up as a sponsor in the future, please introduce them to us. Next. So let's jump right into session 1, which is gene targeting approaches for SDS. I will first provide a brief overview or introduction of the key concepts that will be important for you to fully engage with the talks that will follow. Next. So first, I wanted to introduce again the central dogma of biology, which is that the information encoded in our DNA flows through transcription. So the DNA gets transcribed into RNA first, and then processed into a messenger RNA. Probably everybody by now heard about messenger RNA in the context of vaccines, but this is really at the center of biology in our life every second of the day. And then the information encoded in this messenger RNA is translated into proteins. Proteins are long chains of amino acids that fold up into a three-dimensional structure. And those folded proteins are that's what does all the work in our cells, in our body, from structural component to enzymes to pretty much everything that's necessary for life. And one gene in the DNA encodes one protein pretty much. So that's how the information flows from DNA all the way to the protein. Next. So this is a little more technical, and I'm not going to go into great detail. But so the information in the DNA, so the structure of the DNA is a double helix. That's the first row of this figure. And first, it's transcribed into RNA. Now, it turns out that we don't need the entire sequence of this stretch of RNA to make the protein. We only need the sections in this figure that are highlighted in different colors, and everything in between, the introns, we don't need. So they are spliced out. They are removed in a process called splicing. And that's what generates the messenger RNA from just the pre-RNA or the RNA molecule. And I'm only mentioning this because splicing is important for our particular gene as well, because a lot of the mutations, or actually all SDS patients who have mutations in the SBDS gene, one of their mutation is almost always a mutation at this at exon 2, the junction, so it disrupts the splicing. But this is also important because there might be therapeutic opportunities that can affect the splicing process specifically. But so I'll leave it at that for now. Next. So back again to the central dogma in biology. We have the RNA, the DNA, RNA, and the protein. And how can we fix SDS is the question. So SDS is a genetic disorder. There is a mutation, usually just a single base pair change in the DNA that causes all the trouble. So we could think about fixing that particular typo in the DNA as a therapeutic. Next, we could think about, is there anything we could do on the messenger RNA level to make sure it is actually spliced properly? Or could we possibly affect the next step, which is the translation from the messenger RNA to translating into protein? So our first topic today will actually focus on this third option, on the translation step. The messenger RNA step, the one before that, that could be antisense oligonucleotides. We're not going to talk about that today, but there may be opportunities in that area in the future too. There have been some results published from labs, but currently the technology to deliver it to the right cells in the right amount at the right time is not quite there yet. So we're not going to focus on this one today, but we will focus on both DNA, addressing DNA changes, and also the translation. Next. So just again, I wanted to introduce a few more concepts so that the next talk will be easier to follow. The messenger RNA is not just a random sequence, not just a coding sequence. So in this figure, it's highlighted in, or it's encoded in the middle as a turquoise color. So that's the coding sequence. That is what's going to be translated into a protein. But the messenger RNA also has a start codon and a stop codon. So it has specific sequences at the beginning, at the end, to tell the ribosome where to start and where to stop. In this figure, the ribosomes are colored in green. So they come in two subunits and they dock onto the messenger RNA and start to build these polypeptide chains that then will become the proteins at the end. So what I'd like you to take away from this figure is that there are such things as start codons and stop codons, so certain sequences that signal to the ribosome that their job is done and they should fall off and start over and make a whole new protein when they're ready. Next. So there is a drug that has been developed for something else, but Dr. Valentino Bezzerri's group has been trying to see if it could help SDS patients as well. And just in very general terms, how it works. So on the top, you see the normal translation process. There is the messenger RNA, and there are all these little ribosomes moving along and building the protein. Now, next, with certain mutations in SDS patients, so only some patients have this type of mutations, but in some patients, the mutation can make the messenger RNA sequence appear as if there is a stop signal in the middle. It's not exactly the same as a normal stop signal, but quite similar. And so when the ribosome encounters that mutant sequence, it thinks it's time to stop and falls off and stop translating. So it is not creating the whole polypeptide and is not generating protein. Next, please. So drugs have been developed that help overcome this, this type of stop signal. And in some diseases, they have been shown to allow the ribosomes to read through the stop codon and continue the translation and make protein. So that's the principle of how read-through drugs like Ataluren work. But the next speaker, Dr. Bezzerri, will be able to probably explain it in much more detail and the rationale why they decided to try it for SDS. Next. So with that, it is my pleasure to introduce Dr. Valentino Bezzerri. He worked with Dr. Marco Cipolli for many years at the Cystic Fibrosis Center in Verona, and recently moved to Rome, another institution in Rome. And they have been working on Ataluren for SDS for a number of years and recently published a new article about it. So he's here today to share with us the new findings.
Hi, everybody. Thank you, Eszter, for the kind introduction. Can you hear me?
Yes, we can hear you and see you.
Okay, so let me share my screen. Okay, are you properly seeing my presentation?
Yes, looks great.
Okay, so as Eszter said, I'm actually working at Rome in Link Campus University, but I'm still working as a head of the laboratory at the Cystic Fibrosis Center in Verona, coordinated by Dr. Marco Cipolli, who is present in this discussion. And so briefly, Shwachman-Diamond Syndrome is ultra-rare disease. If compared with cystic fibrosis, for example, where 1 in 2,000 newborns are affected from cystic fibrosis, in SDS, only 1 in 150,000 newborns are affected. The genetic cause is the loss of SBDS expression. The gene SBDS is mainly involved in SDS, even though other genes have been recently associated with SDS, including EFL1 and under investigation are other 2 genes, SRP54 and DNAJ21. Why? Okay. The SDS is affected, primarily affects pancreas. So this disease is featured by pancreatic insufficiency, bone malformation, and mostly bone marrow failure associated with neutropenia, and in many cases is associated with thrombocytopenia and anemia. The molecular defects involves ribosomes, as Eszter said, and the loss of SBDS expression impairs ribosome biogenesis, as excellently explained by Alan Warren Group in UK. And this ribogenesis impaired promotes the impairment or impaired the synthesis of the whole protein amount in the cells. Why we choose the repurposing of Ataluren in SDS? Because we know that the international cohorts of patients with SDS carrying SBDS mutations have a specific nonsense mutation in their genome, namely the 183, 184 TACT. Let me highlight this point. So this mutation is carried by almost 56 or 55% of patients worldwide. And this is a peculiarity of this disease, because if we think at cystic fibrosis, for example, the number of nonsense mutations are much higher. So there are many nonsense mutations with different sequences, whereas in SDS, this is the one of the most present mutations, and is a nonsense mutation. Nonsense mutations promotes the insertion of a premature stop codon in the reading frame of the mRNA. So the mRNA is stopped at a certain point by a premature stop codon, and the resulting protein is truncated, and so is non-functional. If we use Ataluren, this drug may promote the insertion of a substitution in premature stop codon with another random amino acid, so allowing the synthesis of a full-length protein that can be both functional or not functional, depending on the insertion promoted in the protein. Another point that forced us to use Ataluren as a therapeutic option is that Ataluren has already undergone clinical testing in both adults and children with DMD and or cystic fibrosis, showing a favorable safety profile. And it has been tested in thousands of patients. So there are also real-world data that we can use for the safety profile. And we initially tested this drug in vitro to promote the full-length synthesis of SBDS in lymphoblastoid cells obtained from SDS patients and from peripheral blood mononuclear cells. We observed that Ataluren can promote a partial restoration of SBDS full-length protein synthesis, and this restoration allow improved myelopoiesis ex vivo in bone marrow progenitor cells cultivated for 14 or 21 days in colony assays. So we measured the number of myeloid and erythroid colonies after treatment, and we observed a significant induction in the number of these colonies in a small cohort of patients. Given these results, we have extended the preclinical results in an extended cohort of patients, and recently we reported that Ataluren, besides improving the myelopoiesis and the protein synthesis of SBDS, can improve the maturation of ribosomes, so the basic defect of the pathology, and this improvement in ribosome maturation is associated with a normalization of P53 levels. P53 is an oncogenic protein that is highly expressed, upregulated in inherited bone marrow failure syndromes, including SDS, Fanconi anemia, and Diamond-Blackfan anemia. This reduction is not a reduction compared to healthy donors, but is a normalization compared to the pathogenic levels of P53. So we are thinking that it is a good result, not an alert for this disease. Again, we observed that ex vivo, we captured bone marrow progenitor cells with Ataluren, and we observed the maturation of neutrophils, and we improved the maturation of neutrophils, the purple cloud here, after treatment ex vivo. So, given these promising results, we talked a lot with the company owner of the drug, PTC Therapeutics, based in New Jersey, and we reached an agreement for a compassionate program, even if Dr. Marco Cipolli was requesting a registered clinical trial, but due to the agreement, as per agreement with the company, we were able to generate a compassionate program in only 3 patients treated for 1 year with Ataluren treatment, with Ataluren drug, and these patients, for the inclusion criteria, needed to don't show myelodysplastic syndrome, nor acute myeloid leukemia. They were not under G-CSF treatment, and the dosage used was the same used already in clinical trials for cystic fibrosis and Duchenne muscular dystrophy, namely 40 milligram per kilo per day. After treatment, we measured the protein levels as already done in vitro in bone marrow of patients. And you can observe here the general band of SPDS in a healthy donor matched for age with the patients where the protein is very present in the extracts, and the protein in patients before treatment that is very, very low. After 6 months of treatment, the protein levels were comparable with the healthy donors. I repeat, this is not indicative of functional protein. This is just still full-length protein, but is indicative of the read-through of Ataluren. In order to assess the functional improvement, we checked the colony assays as performed ex vivo in our previous reports. And if you look at this figure, this is a healthy donor, how an healthy donor produces colonies, myeloid and erythroid in colony assay with a large number of spots that are colonies. This is a classical SDS sample with hypocellularity and defects in replicative cells, so the colonies are very, very poor. After treatment for 6 months, these are the colonies observed in UPN 26 and UPN 58, namely 2 out of 3 patients, but the third patient is reported in the paper is very similar to these results. And we observed so the Ataluren is able to improve colony formation after 6 months of clinical administration. More importantly, we observed that in bone marrow biopsies, if you observe the upper lane of each patient represent the before treatment 0 month, and the second lane, the lower lane represent the 6 month treatment with Ataluren. We observed an improved cellularity and myelopoiesis, maturation of myeloid lineage after treatment in all 3 patients tested. You can also observe here how poor is bone marrow before treatment in number of colonies that are purple spots, and after treatment how many spots are present compared to the baseline. So we were very, very happy of these, but we checked also the immunophenotype, namely the maturation of neutrophils in bone marrow. Before treatment is very, very low. If we can observe UPN 26, only 20% of cells are mature neutrophils. After treatment, we doubled this percentage, more than doubled this percentage. And so we observe also improved maturation of neutrophils, so functionality basically of neutrophils. You can observe the yellow cloud here, that is 22% of neutrophils, mature neutrophils before treatment, and 64% after treatment. But we were surprised from an unexpected result in pancreas because both fecal elastase 1 measured in stool samples of patients and serum amylase observed in serum sample of patients confirmed that pancreatic activity is restored after treatment in all 3 patients after 12 months of treatment. Importantly, we are trying to recapitulate the same workflow, the same path used for the development of Ataluren in SDS, also for Fanconi anemia, another inherited bone marrow failure, and the preliminary results were very promising, and we recently obtained a fund from Fanconi Cancer Foundation to carry out our research. And this may be important also for SDS community, since an enlargement of the number of patients receiving the treatment or potentially involved in this treatment may improve also the visibility of this drug and force the system, the health system, to promote an extension of these studies for SDS. So the take-home messages are that we attempted the first study of precision medicine approach for SDS, and more broadly we can say that it is the first clinical attempt for inherited bone marrow failure with the precision medicine approach. Our clinical results are prompting us to move forward to an international clinical trial, or at least to request to the FDA and EMA agency what they are requesting for move forward in clinical development. The limitation of this study is the small sample size, but we have to consider that, as I said, SDS is ultra rare. We can treat only 55% of our patients, and in these 55% we have to exclude the people with bone marrow transplantation, and with people with myelodysplasia, and people receiving GCSF. So 3 patients is not a large number, but for this condition is indicative. This study strengthened the actual clinical benefits of Ataluren, since this drug has been discontinued also for Duchenne muscular dystrophy due to the variability in clinical efficacy demonstrated during the years. In our case, we are postulating some explanation of the actual efficacy of Ataluren for SDS, since we are treating a proliferative tissue as bone marrow, and with only one nonsense mutation that seems to respond very well to the treatment. And these are peculiarity of SDS that may justify the clinical benefit observed. Our findings lay the groundwork for repurposing translational read-through, not only for SDS, but also for inherited bone marrow failure syndromes, including Fanconi anemia and the Diamond-Blackfan anemia. Let me say that I am the presenter of this study, but the study was co-conceived by Dr. Marco Cipolli in Verona, and I worked with his group for many years, and I have to thank all the young researchers involved in the study that were very hard worker for carry out these results, and also to all collaborators both in Italy and in US that helped us to reach this milestone in SDS treatment opportunity. These are the findings received for carry out these studies from 2014 to date. And thank you all for your attention. I'm ready for your questions.
Thank you. Thank you, Valentino. This is really exciting. Drug repurposing has a lot of benefits in terms of the speed with which things can get into patients, because the safety has already been established, manufacturing, all the things have been already established. But as you mentioned, there are still other types of challenges that come along with that. So we are ready now for questions from the audience, if there are any. Please type them into the QA box. Let's see. One question. there's a question about timing from Jackie. When will you proceed to a larger cohort trial?
Oh, it depends on the request from the medicine agencies. For ultra-rare diseases, the rules are different from the other diseases. We cannot think to design an enlarged study with hundreds of patients because it's not realistic. We have to design a very small clinical trial, or try to convince the agencies, FDA and EMA, to design something different. But also we have to try to produce the drug or have the drug from the company. There is in this discussion also Dr. Marco Cipolli for the clinical issues of development that are not my actual field. I don't know if Marco want to add something to this discussion, and but we are trying to proceed in the best path to accelerate the clinical development. That is probably not only a registered clinical trial.
Yeah, that is related to another question from the audience about what concrete steps regarding clinical trials are you thinking about? Don't know how much you're able to share, but that's the question in the chat.
Yeah, so the question is about GCSF.
No, there's an earlier one just asking about what concrete steps might take us to actual clinical trials, whether you're able to share any specifics about that.
I don't know. We need the drug. We need the drug because the drug is patented by PTC Therapeutics. They provided the drug for 1 year under an agreement, international agreement. The agreement is now stopped. The results are published. Now we have to do another agreement with PTC Therapeutics or proceed with other paths that are still under evaluations, both in US and in Europe, that have a different agency for the approval of the drug, and it is a big effort from the clinicians now to understand what are the best steps to accelerate the translation to the small cohort to enlarge the cohorts of patients.
Yeah, so it sounds like this is not so much a scientific question, but more around regulatory and business and all the other things that go into drug development.
Yes, regulatory mostly, because EMA and FDA are not the same, have not the same rules.
Yeah. And then the other question is about one of the attendees heard you say that patients who are on GCSF would be excluded. Does it apply to patients who are currently on GCSF? Or yeah, can you say more about that?
Yeah. No, we excluded the patients under GCSF because if we have to observe myelopoiesis and the improvement in neutrophils number or platelet number, we know that growth factors as a GCSF per se promote the cellularity, so they will be disturbing inputs in the patient and tends to destroy the study. So people under GCSF are excluded from these studies.
Yeah, and I understand GCSF is also, the effect of G-CSF also fluctuates strongly even during the day. So it sounds like the reason you're excluding patients who are currently on G-CSF is because it would really disguise or change the data that we would want to observe just based on Ataluren itself.
Yeah.
But if somebody took G-CSF years ago, that's fine. They just shouldn't be on it
That's
right
fine.
now.
Yes, the effect of G-CSF is transient. So once you stop the G-CSF and the blood has a washout of this drug, there are no problems to take the drug after washout of the G-CSF.
That's good. So that's all the questions I see in the chat. Unless Dr. Cipolli wants to hop on and say anything. If not, we will jump to the next topic. So any last questions or comments?
Marco, any news?
Okay. Do you listen me?
Yes, we can hear you.
Yeah. Oh, news and the next step is really not easy because, as Valentino already explained, we have two or three aspects that we have to discuss. Also because we at the end published just the article arrived just last month, so our possibility to discuss with companies interested to develop this way, or the regulatory agency is just it was linked to have a publication about that. So one aspect is that the patent for the fabrication of the drug expired last January. So PTC practically stopped to produce, but it's possible to now that it's possible to produce as a general drug. I don't know if in US is the same, but probably yes. So this is one aspect, and we have to consider if there is a company that is interested to develop and to produce. The other is to discuss with, in this case, with the FDA first, because the patent expired in US, but not in Europe still. In Europe, it will expire the next January 2027, January 2027. And so our point is to discuss with the FDA to understand what they consider for the developing of the study at the end. As Valentino explained, it's not possible to do a real trial with blind section because we need many patients for doing that. But it's also true that we have a very clear outcome. So our principal outcome is very clear, and is the effect on the bone marrow. So it's not a secondary outcome, but or a surrogate outcome, but is a real outcome. Is exactly we measure what the drug does. So in this case, we have experience in CF that FDA approved the modulators for CFTR also on the basis of biological demonstration. So on cellular models is ex vivo, but cellular models. So if we consider that, one possibility is to ask if it's possible to start giving to the patients the drug under evaluation. So in the sense that we can start during a precise timing, we have to describe what is happening. for example, in Europe, the EMA decided this strategy for the Duchenne. And after some period, some 1 year, 2 years, the company needed to show the results. And so under this way, the drug has the possibility, or had the possibility to be done to the patients. Now EMA decided to stop because there was no evidence of efficacy for Duchenne. But here we are in a different field, because for example, I don't know if you understand that, but the outcome to evaluate the efficacy of the Ataluren in Duchenne was the 6-minute walking test, or the walking test in general. So you understand that this is not a direct evaluation of the effect of the drug. We are discussing with the company that now they seem to be interested to help us to develop that. And the next step, I hope in the first month of the next year, is to discuss with the FDA to understand how they can suggest to us to proceed in our work. So because FDA can also give us suggestions, and just before to say yes or no. And so this is our next step. The other step is that we already are working about that, is to show what it means to stop the drug in these patients. So we are evaluating bone marrow after 6 and next month after 1 year from the end of the therapy. We already have some results, but not all. So, but we'll of course will publicate also all these data. And I hope that in spring probably we already finish also this part.
Yeah, thank you. Thank you. This is so fascinating to me that in some ways the science seems to be the easy part, and everything else is hard and complicated. I also find it really interesting to follow what happens after therapy, whether the effect remains or get worse after the drug is stopped. I think that will be very informative to inform the next steps too.
We are following up the patients for this reason.
Mm-hmm. Yeah, so nobody else typed in questions. Yes? Somebody was saying something? Yeah, so thank you so very much for joining us today. So super interesting. If the audience sends us more questions later, I'll email you and we'll may be able to.
Okay, sure.
To
you can write me. Okay. Bye-bye. Okay. Have a good afternoon. Thank you.
All right. So our next topic is, again, other types of approaches to possibly treat SDS. If we go back to our central dogma slide, we will now switch focus on the DNA. So SDS, as we all know, is a genetic disease. So it results from mutations in the DNA sequence. So very naturally, we would be very interested in fixing the problem right there. Just a reminder, all of us in all our cells have 22 pairs of chromosome plus XX or XY. And the DNA is packaged up in this chromosome structure. But if we unravel it, we would see the double helix. And in the double helix, we see base pairs. So bases are chemicals, molecules attached to the backbone of the DNA that pair with each other. And if there is a change to those structures, then the sequence changes. And if the sequence changes, the amino acids that are encoded by those bases will change as well, and that's what results in problems. So another concept that I wanted to introduce in order to make the following talks more familiar or understandable is the difference between ex vivo and in vivo therapy. Ex vivo means that the therapy is happening to some extent outside of the body. You are all familiar with bone marrow transplant. That is not drawn out here, but that is basically a cell therapy too. So in a bone marrow transplant, a donor donates their hematopoietic stem cells. It's cleaned and filtered, and then it's infused into a patient after conditioning, after the bone marrow niche has been emptied to make space for those new cells. So that's our standard bone marrow transplant or stem cell transplant that all of us have heard about. Now for ex vivo gene therapy, it's kind of a similar process, but the difference is that the cells come from the patient themselves. So the patient donates their stem cell, so to say, it's filtered, and then the gene in those stem cells is fixed, repaired, and other, there's filtering and expansion, like other things happening to those cells, and then the cells are infused back to the patient. The therapies you may have heard about so far for sickle cell disease, that's how it works, and it does include conditioning. So in sickle cell, hematopoietic stem cells are taken from the patient. The genetic editing happens to the cells. In the meantime, the patient receives conditioning regimen to get rid of the old bone marrow, or at least create some space for the new cells, and then the new cells are infused back. For our purposes, there are some thought that maybe the conditioning could be reduced or eliminated in such a process, but we'll talk about that a little more later. And so in contrast to this ex vivo approach, there is in vivo approaches in development as well. In that case, instead of taking cells out of the patient and fix them outside the body and then put them back, in the in vivo approaches, the editing of the gene happens inside the patient's body. So what would be infused in a patient in that case would be a vector that, or a particle that includes the CRISPR and other components, and the editing would be happening in the body. And so there's a lot of hope in this brand new field that it could help eliminate a lot of the side effects that come from the conditioning. So it could be maybe done outpatient, and it might be just overall easier on the body, and it may be also cheaper, less expensive. Classic bone marrow transplant or gene therapy transplant are very, very expensive. Like even the classic bone marrow transplant is probably about $1 million in cost, and with gene therapy, it's even more. So I just wanted to introduce these terms to you so that the following talks will be easier to follow. And with that, I'm very excited to introduce Dr. Dan Bauer's talk. Now, last week has been an extremely busy time for people involved in gene therapy because there were 2 big gene therapy meetings in Spain, plus there were deadlines for big NIH grants. So unfortunately, Dr. Bauer wasn't able to join us in person today, but we will show a recording of his talk. And we have a colleague of his in online too, and his postdoc. And so we will be able to have a discussion afterwards about this line of work. So Dr. Bauer's talk will share the prime editing on the hematopoietic stem cells as an ex vivo gene therapy approach.
Hello, my name is Daniel Bauer. I'm a pediatric hematologist and physician scientist at Boston Children's Hospital. I'm going to talk today about opportunities for therapeutic gene editing for Shwachman-Diamond syndrome. So in hematology, we actually already have a standard curative approach for blood disorders, and this is bone marrow transplant, and this is where we can replace the blood system of an individual who has a blood disorder by transplanting hematopoietic stem cells from a healthy donor. And these are the stem cells that make blood cells for life, and so this can replace the blood system of a person who has a blood disorder and cure their disease potentially. But this has challenges, and this depends on a donor pool that's limited. We need to find the best matched donors, and that can be difficult. And even with a matched donor, there remains some incompatibility between the immune systems of the donor and the recipient, and that leads to risks such as graft rejection and graft versus host disease that make the procedure more complex. And there is some morbidity and even mortality associated with this procedure. And our alternative approach could be to collect the hematopoietic stem cells from the patient, him or herself, to modify them to fix the problem of the blood disorder, and then to reinfuse those cells to replace the patient's blood system with a blood system that no longer has that blood disorder. And how can we do that? Well, we can take an approach known as genome editing. And here's an artistic depiction. And the idea is we can make precise changes in the DNA sequences that are associated with disease in living cells. And then we can fix the potential of those cells to restore health. And if we do that in hematopoietic stem cells for blood disorders, those are the cells that produce the whole blood system, and they can overcome the blood disorder. And so how do genome editing technologies work? Well, there are these amazing molecular machines that have basically two parts. There's one part that's for DNA targeting, and you can imagine if you'd use an analogy of sending a package, this is like the address, the street address, and the zip code that gets that package to just the right place. And then there's the contents of the package, which are the DNA modifying parts that once the genome editing tool has gotten to just the right place in the genome, it can make a change. And if there's a mutation that's causing a blood disorder, they can potentially fix that mutation. And so genome editing today is based on engineered CRISPR systems. And CRISPR systems are actually naturally occurring immune defense systems that bacteria have to fight off viruses that infect those bacteria. And this is showing the structure of CRISPR protein wrapped around RNA, shown here in orange, and then DNA, shown here in blue. And so you can see how the RNA and the DNA make this tight connection. And here's kind of a cartoon version of this, where normally the DNA would be double-stranded and have this blue strand and gray strand. But now the RNA, shown here in orange, can invade and target just that specific sequence of DNA that matches the orange and blue match here. And now, once that locks in, the CRISPR protein can engage, and these two triangles show these cut sites where the CRISPR protein can make a change in the DNA, and that can be a permanent change with a one-time CRISPR exposure. And here's kind of an artistic vision of like the universe of DNA in the nucleus of a cell, and in a two-part recognition mechanism, the CRISPR system first finds these kind of yellow docking sequences, and then this red RNA sequence, if it matches the target DNA, it can unzip the DNA, zip in the RNA, and allow the CRISPR molecule to engage and make a change in the DNA. And what's so powerful about these systems is just by changing the RNA sequence, we can retarget the CRISPR system to essentially anywhere in the genome, so we can target almost any DNA sequence we want to by making a simple modification of the design of these CRISPR systems. Now, when we think about gene editing, it's really important to think about the precision of the approach. We want to target where we want to target, but nowhere else in the genome. Imagine these are two chromosomes, a blue one and a gray one, and there's a, let's say, a place in the blue one we want to target, and this is like in green showing the intended edit, but we want to avoid all these unwanted possibilities, like some other edit at that region, or targeting another chromosome, or making a deletion, or a translocation of the chromosomes, or loss of chromosomes, or shuffling of chromosomes. So we need to be very precise when we're doing this to make just the changes we want to make and no other changes. And we need to deliver these molecular machines into cells, right? They're no good unless they're actually in the cells of the patient, where they can fix the problems associated with the disease. And so we have all sorts of ways to deliver these gene editing systems into cells. We can use electrical energy through electroporation. We can use physical means with mechanical poration of cells. And we can use biochemical and nanotechnology methods where we can engineer particles that can deliver CRISPR systems into cells. And so we have all these options available to us. We did extensive study of the precision of the approach. Without going into detail, we learned a lot along the way. And one of the key findings was that genetic variants that we all have that differ between us can create risks of off-target effects. And we found a common genetic variant that could produce an off-target potential for the initial version of the gene editing therapy. And so we use that information to design an even more precise therapy. And we developed a new method to look for these off-target effects that was very useful in allowing us to get FDA approval of our therapy, and I think is kind of a new standard in the field to look for genetic variant associated off-target potential of gene editing. So with all that background, what are we doing now? We're working on therapeutic gene editing for Shwachman-Diamond syndrome. So as you know, Shwachman-Diamond syndrome is a genetic condition where the blood disorder component is a main risk for morbidity and mortality, and that there's a mutation that almost all patients carry in the second intron of the SPDS gene, which greatly reduces the splicing of the gene and the production of a healthy SPDS gene. And why this is a favorable feature from the standpoint of gene editing is that we could design a universal editing strategy to fix this mutation that could then be applied to almost every patient with Shwachman-Diamond syndrome. In addition, the disease itself is associated with decreased fitness of the hematopoietic stem cells, and there's frequently compensatory somatic mutations that are observed, which suggests that a relatively low threshold of editing could provide great clinical benefit. And we have proof of concept studies underway, and we have very encouraging preliminary results that we can get great efficiency and precision of gene editing for Shwachman-Diamond syndrome. So our vision is to develop an investigator-initiated clinical trial of therapeutic gene editing for Shwachman-Diamond syndrome. And I should say this is a collaboration with my colleague, Dr. Akiko Shimamura, who's also at Boston Children's Hospital and who leads the SDS registry. And so Our next steps are to complete our proof of concept studies, to do off-target analyses and safety studies, obtain clinical grade editing materials, develop our cell manufacturing process and our clinical protocol, all to achieve regulatory approval so we can initiate the clinical trial. And we have a track record of doing this, and we kind of know the next steps to proceed with. So thank you very much for your attention. And I'm really excited to further develop our approaches for therapeutic gene editing for SDS.
Yeah, so this is a super exciting project. And one aspect that we heard in Dr. Bauer's talk were the off-target effects. So that's where the gene editors would make changes that we don't really want them to do, and how to think about those and measure those. That's the focus of Dr. Tim Barry's work. He works closely with Dr. Bauer. He's a postdoc in Dr. Bauer's lab in collaboration with several other labs, actually. And we've recently nominated him for an Uplifting Athletes Youngest Investigator draft, which is a matching grant. And we look forward to hearing about whether it was successful or not in November. But in the meantime, we recognize the importance of this work and look forward to working with Tim in any capacity. So on to you, Tim.
Okay, thank you so much, Eszter. Am I audible? Can you hear me?
You are audible, and your slides came back up, but not in presenter mode yet.
Okay, sounds good. Let me hide the video panel. Okay, hopefully it will be visible in just a moment.
Yeah,
okay.
Looks perfect.
Okay, so yeah, thank you so much, Eszter, for the invitation to speak here. Yeah, as Eszter mentioned, I am a postdoc in Dr. Dan Bauer's lab. I am the statistician in the lab, so my background is not in biology. It's in math and computer science and statistics. And yeah, I recently became interested in gene editing approaches for treating rare diseases, including SDS. So yeah, I'm excited to be here to share some of our work, and I look forward to learning from other scientists who work in this area, as well as from families, to learn more about the human side of the condition. But yeah, so the focus of this talk is on off-target effects in these therapeutic gene editing approaches. So as we just heard from Dan, gene editing is a promising approach to treating SDS. However, as I will describe, gene editors can sometimes install unwanted mutations in cells. Not at the sort of target location, but at off-target locations in other parts of the genome. And these unwanted mutations can present safety concerns. So the goal of the project that I'm working on now is to deeply characterize the editing profile of editors relevant to SDS. This will enable us to better understand the safety profile of these editors and ultimately accelerate therapeutic development for SDS. So yeah, so we learned from Dan and Eszter, how these gene editing approaches work. But just to briefly summarize, the majority of SDS patients harbor the same mutation. So there's a particular nucleotide that's very close to exon 2 of the SBDS gene. And in most SDS patients, this nucleotide, which is a T, is converted into a C. And this mutation, this substitution reduces activity of the SBDS gene. And so the idea behind gene therapy approaches for SDS it's a sort of conceptually simple idea. SDS gene editors would convert the C back into a T in SDS patients. And so this would sort of fix the mutation and hopefully rescue normal cell function. And so a major focus within the Bauer lab is on prime editing approaches for therapeutic gene editing. So prime editors are an advanced sort of version of CRISPR editing that enable us to make single nucleotide changes, right? So sort of we can edit the C and convert it into a T without sort of altering, without altering the nearby letters. So, at a sort of high level, that's the idea. And as Dan discussed, okay, so we have two approaches, ex vivo and in vivo. Just to sort of recapitulate what Dan said, in the ex vivo approach, we collect patient stem cells, maybe from blood or from bone marrow. We use CRISPR editing to alter the DNA of those cells, and then we infuse those cells back into the patient. A sort of newer and very exciting approach to therapeutic gene editing is the in vivo approach, where the little machine, right, the CRISPR editor that makes the edit to the DNA is delivered directly into the patient's cells, and then the editing happens within the patient's body. And so, what I'm going to describe sort of applies equally to ex vivo and in vivo gene editing approaches. Gene editing for SDS is promising, but several major challenges remain before this sort of technology comes to fruition. In the context of in vivo gene editing, for instance, we need to be able to deliver the gene editor to the sort of correct cell type within the patient. We need the editor to efficiently edit the SBDS gene. Also, we need minimal off-target editing, right? So I'll be describing this sort of over the rest of the talk, but we want the editor to not make too many edits at sites that are separate from the on-target site. And of course, the cells that have been edited need to survive and proliferate within the patient, right? So, so a number of challenges. And the major focus of this talk is on challenge 3, namely off-target editing. So one challenge of these CRISPR editors is that they can install unwanted edits at off-target sites. So as Dan mentioned, the way these CRISPR editors work is we basically program the primary sequence of DNA that the CRISPR editor is supposed to identify into the CRISPR editor. It's like an address, and then the CRISPR editor sort of makes an edit at that particular location. But a challenge here is that if there's another location in the genome with a very similar primary sequence to that of the on-target site, the CRISPR editor can go to that location of the genome instead and make edits there. So, just by analogy to, Dan's, or to extend Dan's analogy of, sending mail, right? Like if we send mail to a certain address, maybe it sometimes is delivered to another house, where the address is like very similar, right, to the house that we intended to deliver the mail to. And so, of course, if there are mutations that are happening at off-target sites, this is potentially harmful, right? Off-target mutations could lead to cell death or cancer. And so we want to minimize these off-target editing events. And so a major goal of therapeutic gene editing in the context of SDS, and this applies more broadly to other rare diseases, is to understand, what are the editing sites of clinically relevant CRISPR editors. So here we're looking at, the entire set of chromosomes. We know that the SBDS gene is located on chromosome 7, so I've got a blue dot there, right, indicating presence of the SBDS gene. And in red, I have example off-target editing sites, right? So for a particular CRISPR editor, right, maybe hopefully it's making edits at the on-target site, right, which is indicated by the blue dot. But the CRISPR editor might also be making edits at these various off-target locations, right, that are shown by the red dots. And so an important step of therapeutic gene editing is to understand the editing profile of these therapeutic CRISPR systems, right? At what precise locations in the genome are these CRISPR systems making edits? Looks like we might have something in the chat. Okay, so right, so understanding the off-target editing activity of therapeutic CRISPR systems can help accelerate the translation of gene editors from lab into clinic. So just to give you a sort of example of why we would like to understand the off-target editing activity of CRISPR editors, right? So suppose we are evaluating 5 candidate gene editors for SBDS, right? So we might have 5 candidate gene editors shown here in different colors. Maybe these gene editors are all targeting the same location, but they might have slightly different chemical properties, leading to slightly different off-target editing profiles. And so if we can, characterize the off-target editing activity of these CRISPR editors, we can then select the one that is best, right? In the sense that we can select the one that has the lowest, amount of off-target editing, right? So, in this example, maybe we want to pick candidate editor 5 here, right? Which has minimal off-target editing in comparison to these other candidate editors. And also, profiling the editing activity of therapeutic CRISPR systems is important, is an important aspect of obtaining FDA approval for these therapeutics. So several assays have been developed to measure the off-target editing activity of CRISPR editors, including assays like GuideSeq and PE-tag. So, we won't get into too much depth in this talk about how these assays work, but I just want to describe at a high level sort of what the idea is. So, these are assays. So these are preclinical assays, right? So we're conducting these experiments in the lab as part of the therapeutic development process. So the first step is to collect DNA, ideally from an SDS patient. And so once we have this DNA, we sort of put the DNA into a solution. And then we add to this solution the CRISPR editor as well as a molecular tag. And the molecular tag is going to basically tell us where in the genome is the CRISPR editor making edits. So the CRISPR will edit the DNA, right? The CRISPR will edit the DNA, hopefully at the on-target site as well as at some number of off-target sites. Sorry, there was a little bit of feedback. I'm not sure there's a question, but the CRISPR editor will edit the DNA at the on-target site as well as at a number of off-target sites. And at all of the off-target sites at which the CRISPR editor makes an edit, this molecular tag will be inserted into the genome. And the final step is to do DNA sequencing, right? So we just sequence the DNA, and by sequencing the DNA, we're able to ascertain where these molecular tags have been integrated and thus where the CRISPR editor has made edits. So, just to give an illustration of what some of the data look like. Oops. So these are data from a GuideSeq assay that we conducted in the Bauer lab. The different panels here correspond to different chromosomes. Okay, so I'm showing 20 chromosomes in this figure. And the x-axis of a given panel indicates the genomic coordinate of that chromosome. And the y-axis indicates the sort of the number of reads, right? The number of editing tags that we've sequenced at that particular genomic coordinate. And by looking for peaks, right? So we see here there are several clearly defined peaks, right? So by looking for peaks in these data, we can identify the regions of the genome at which the molecular tag was integrated, and thus where the CRISPR editor is making edits. So sort of that's the high-level idea of these assays, right? That aim to identify, sort of the that aim to pinpoint where the CRISPR editor is making edits in the genome. And there are variants of this assay that are applicable to CRISPR-Cas9 editing, as well as sort of newer assays that are relevant to prime editing. And so, as I mentioned at the beginning of this talk, I'm not a biologist. Okay, I'm doing my best, to learn the biology, but my background is in statistics. And although we have several assays that have been developed for profiling the editing activity of CRISPR systems, unfortunately, there does not yet exist a comprehensive statistical and computational framework for analyzing these data. And it turns out that these data pose several statistical and computational challenges that are pretty non-trivial. And so the goal of my work is to develop a unified statistical toolkit for analyzing these CRISPR editing data. And as part of this effort, the Bauer lab is generating a large amount of gene editing data related to SDS, and so we're going to be testing out the statistical and computational methods, in large part on data produced for SDS gene editors. So that's what I wanted to talk about today. So in summary, the sort of one-sentence conclusion is as follows: characterizing the off-target editing activity of SBDS gene editors will accelerate the translation of therapeutic CRISPR systems for SDS from lab into clinic. So with that, I want to thank my three PIs, Dan Bauer, Luca Pinello, and Danilo Pellin. Thank you so much.
Thank you, Tim. So we can launch right into a short Q&A session. As mentioned, Dr. Bauer wasn't available to join us, but one of his colleagues, Kit Shaw from Boston Children's Hospital, is able to join us for a discussion. She is Director of Clinical Translation and is closely involved with the SDS project. And she has depth of experience with translating therapies from the lab into the clinic, working with the FDA, and that sort of line of work. So please, everybody, if you could put your questions in the Q&A. I don't know if Kit is actually online.
Yes, I'm here. Can you hear me?
Okay. Yes, yes. So while we're waiting for more questions from the audience, I was curious, in your experience, Kit, how much of a weight does FDA put on these off-target effects currently? Like, are they considering it to be a big risk when it comes to new therapy, or how does FDA think about risks?
So when we submit a package for the IND, ideally we will have had a pre-IND meeting with FDA where we would discuss the expectations of what they would like to see in an IND package. And in the past, FDA has definitely wanted to see preclinical data on if we've looked at off targets and the risks of where they could possibly go. So definitely in the IND package, FDA will want to see a genotoxicity package describing what we did to look for off targets. They are not so prescriptive as to what assays they require us to do, but they will have expected that we either look using computer models or doing the assays that Dr. Berry discussed, in cells, and to describe what we think the off-target profile will look like.
Mm-hmm. And so was that a similar process then also for the sickle cell therapies?
Yes, that is correct. We, for the sickle cell project, which Dr. Bauer has an IND for, there was a comprehensive off-target package that was submitted to the FDA as part of the IND.
Mm-hmm. Yeah, I wanted to remind the audience too that in Dr. Bauer's talk, the first half or 2/3 of the talk was focused on his experience with sickle cell disease. And so the hope is to draw from all that experience and apply that to an SDS therapeutic as well. So we have a question from the audience, which is, do you have, I suppose Dr. Bauer's team or the SDS project in general, does it have a medical ethicist on the team?
So in Dr. Bauer's team, we do not have a medical ethicist, but what will happen is once we've demonstrated a good preclinical proof of concept for the editing strategy that Dr. Bauer will use, this will all be put together into a clinical trial protocol, which describes how the patients will be treated. And the first iteration, I think, will be ex vivo, as you previously described, Eszter, how that looks like. In developing this clinical trial protocol, we would hope to have patient advocate input into describing some of the endpoints and what is important to the patient community. And when this clinical trial protocol is developed and written, it has to go and be approved through an IRB and also the FDA before any patient can be treated. And so within those regulatory reviews, there typically is a medical ethicist that will, review what the treatment is going to be and provide input. Definitely in the Boston Children's IRB, I'm pretty sure there is a person who sits on that committee as a medical ethicist. So that's how that would work.
Yeah, that makes sense. All right. And then there's a question about the timeline. Of course, how many years do you think before this would be ready for IND submission? Or we could also talk about when do you think the group would be ready for the pre-IND discussion, and then how long would you expect it to take for the IND submission?
So as soon as we have pretty solid preclinical data and different packages within that IND package, which would include proof of concept, as Dr. Bauer explained in his presentation, and then a section also for the off-target, and then the clinical trial protocol written, that's the time when we would go to the FDA with hopefully a pre-IND and kind of ask for, is this sufficient? Would you support if we submitted what we have here for an IND package? So to get to an IND package, that's really difficult to say how long. I would think, based on my knowledge of where Dr. Bauer is with his studies, I think it is conceivable that we could be having a pre-IND meeting, maybe around 6 months or so from now. And then depending on FDA feedback, what they would require for Dr. Bauer's team to do to submit the IND package, it could be anywhere from another 6 months to a year. But I think because Dr. Bauer has experience already with what needs to be done, I do think that it could be a shorter timeline than for some other packages that, maybe somebody who doesn't have experience doing. And also, I do know, because this is a rare disease, that the FDA, in my experience, has seemed to be more open to shortening those timelines, because I think they do realize that there needs to be activity in the rare disease space. So my experience has been that they have been really willing to engage and to discuss how to shorten timelines for developing of these treatments.
And that's including for the amount of time that they spend reviewing things, and then also considering data they might require to make them maybe structure them in a way that can be obtained faster.
Yes, I can't speak for the FDA, but they do, they are bound by certain regulations. Once we submit, a pre-IND package, they are required to have a meeting with us within 3 months, I think it is. And then when we submit an IND package, they are required to respond to us within 30 days. So those types of timelines are not really dictated, by their review. But I guess I'm speaking more of, the clinical trial protocol itself, and to actually get the product into more patients. I think FDA is really working on shortening those kinds of timelines once you have like an IND open.
Mm-hmm. And talking about those timelines and people, do you have a sense of what type of patient would be the first ones to try this type of therapy?
So when the clinical protocol is developed, we do have to come up with a list of inclusion and exclusion criteria. And I personally am not a clinician, so unfortunately I can't speak to what type of patient would be the best for that. But yeah, I mean, I think it would have to be a discussion between the clinicians and the patient advocacy groups to really work on describing those inclusion criteria. Now, the challenge with these types of diseases is actually finding the right kind of patient, because you do have to walk a fine line between making sure that your treatment, when it goes to the patient, is not treating somebody who is so ill that you will not see any benefit. But at the same time, the FDA can be very risk averse, and they will want you to treat a patient who could have some benefit also. So it's really a fine line that is a discussion between, FDA and the clinicians and patients, kind of describing what would be the ideal patient to, have first treated in this type of protocol.
Mm-hmm. Yeah, in other words, like considerations could be, are we looking for adults versus children, or are we looking at people who have no clones that look scary at all, or maybe some clones already, or maybe even people who have already progressed to a certain stage or have MDS or leukemia. So those are the discussions that will need to be had of like who will be included in the first.
Correct. Yes, and because this will be a first in human trial, you can imagine that FDA would really want it to be as safe as possible, and have some kind of benefit, I think, also for the patient.
Mm-hmm. Yeah, I think that will be a really, really important discussion and consideration in the planning process. Let's check in if there are any other questions. Any last questions from the audience? Please type them in now. If not, we can move on. But I really, really wanted to thank you, Kit and Tim, for being available on this Sunday and share your insights with us. This is really, really meaningful to the community. So thank you.
Thank you. You're welcome.
Okay, so I don't see any other questions. So I'll be moving on to the next topic. Let's see. So on the agenda, we were planning to present Dr. Elefanti's work in Australia, but in the interest of time, we will jump ahead to Dr. Sabatino first and cover Dr. Elefanti's talk afterwards. Just wanted to share again this slide to put ex vivo and in vivo therapies right next to each other, because the hope is that if we are able to deliver in vivo therapies in the future, that we could cut out a lot of the really hard parts of the cell therapy, transplant conditioning, that sort of thing. And so that may then lower the toxicity and the time and the cost for it. But this is in development. We are not quite there yet. All right, so I wanted to welcome Dr. Denise Sabatino. She's Research Assistant Professor of Pediatrics at CHOP, or University of Pennsylvania and Children's Hospital of Philadelphia, to be precise. And she will share with us some insights about KJ's story that was all over the news a few months ago, and the trends in gene therapy.
Thank you for the introduction, Esther. Let me see if I can share my screen. Whoops.
While you are sharing your screen, I wanted to mention that the way we got introduced was through the American Society of Cell and Gene Therapy, which is a big society, foundation or organization here in the US that promotes cell and gene therapy development. And they have this wonderful program where they connect patient advocacy groups and other groups to speakers if and when we want to learn about any of these questions. So I'm super grateful for the opportunity to connect with Dr. Sabatino. The website, the American Society of Cell and Gene Therapy's website, also has a lot of educational resources for patients. And I will link to that in the materials that I will distribute after the meeting. So over to you, Dr. Sabatino.
Thank you, Eszter. So as Eszter mentioned, I'm a researcher at the Children's Hospital of Philadelphia. And the focus of my research is developing therapeutics for hemophilia. But as Eszter mentioned, we were introduced through the American Society of Gene and Cell Therapy. And I'm happy to share a story that came out of the Children's Hospital of Philadelphia, or CHOP as we call it, about a patient with a rare disease, a rare genetic disease that was treated with a personalized gene therapy approach. And as Eszter mentioned, this received a lot of press, and I'm happy to share the story that's come out of CHOP. So this is KJ's story. It was about a first of its kind personalized gene editing approach that was developed at CHOP in partnership with an investigator at the University of Pennsylvania. So Dr. Rebecca Ahrens-Nicholas is the clinician at the Children's Hospital of Philadelphia who was the lead on this project. She is KJ's doctor, and she worked alongside Dr. Kiran Musunuru, who is at the University of Pennsylvania, and his expertise is developing gene editing approaches. So together, their teams really moved this project forward that led to this story that I'm going to share with you today. So what genetic disease does KJ have? He has a rare inherited urea cycle disorder called CPS1 deficiency. So these babies are usually diagnosed in the first few days of life because they become very sick. And the reason why these children become so sick is because their bodies cannot properly, excuse me, I'm going to make an adjustment. The reason why they become so sick is because their bodies are not able to break down proteins because they're missing a very important enzyme in their liver. So their livers are not able to remove ammonia. Ammonia is a waste product that our bodies normally make, and it's excreted through the urine. In the case of KJ and children with CPS deficiency, this ammonia builds up, and then because they cannot excrete it, and it can become very harmful, especially to their brain. And this can cause long-lasting damage if it's not treated quickly. So normally these patients are put on a very restrictive diet. They also receive medication to try to manage their ammonia levels. These patients, though, usually need a liver transplant. The challenge in treating these patients is that the patients cannot be candidates for a liver transplant until they have grown large enough, until they are a little bit older to become a candidate. So during that time, about half of these patients with CPS deficiency die because they aren't, it's been very difficult to manage them clinically until they become, until they're able to be candidates for a liver transplant. So finding new ways to treat these patients who are too young and small to receive liver transplants would really be life-changing for families that have children with this disorder. So as we heard by Dr. Bauer's group, gene editing is a new approach that's being developed for many different diseases. And this is the approach that was used to treat KJ. So KJ's DNA had a misspelling in the DNA, or a typo, as Eszter put it earlier, that affected this enzyme that he needs to remove the waste from his body through the urea cycle. And so the aim of this approach was to correct the misspelling in his DNA to help treat his disease. So this schematic outlines the treatment in a little more detail. So the tools to correct the misspelling in his DNA were delivered in a lipid nanoparticle. So the gene editing tool called CRISPR was especially designed to go to the exact site in KJ's DNA where this misspelling occurred that was causing his disease. This drug is delivered intravenously, so it's delivered into the bloodstream, and it travels then to the liver. These lipid nanoparticles are very efficient at targeting the liver. And so this is why, in part, that this disease was a good first candidate for this type of in vivo approach. Once the tools are in the liver, the gene editor called CRISPR enters the nucleus, and it is programmed, as I said, to go to the exact site in KJ's DNA where that typo or misspelling was. And that's where it works to correct the gene in those specific liver cells. The hope then is that the cells can then start making the enzyme and start processing the ammonia, which his body wasn't able to do before. So importantly, KJ received the lowest possible dose of this therapy that could potentially allow his body to then adapt to the therapy, and they are also trying to minimize risks. Another interesting aspect of this work is that because it's delivered in a lipid nanoparticle, it's possible to deliver this more than one time. And so KJ received total of 3 doses of this approach over a number of months. So why was KJ an ideal candidate to be one of the first patients to receive this in vivo gene editing approach? Well, Dr. Ahrens-Nicklas and Dr. Musunuru have been developing the tools to correct the misspelled genes in their teams for over 2 years. And they are developing these in the hope they could be used to treat any patient with a genetic disease. So they had been practicing how to create these custom tools for children, and specifically for urea cycle disorders, which is one of Dr. Ahrens-Nicklas's specialties. When they met KJ, they really thought that maybe it was the right time to try it in a patient. Importantly also, KJ had the most severe form of the disease, so he was at very high risk of complications. So this, they felt, also made him a good candidate. And in this disease, each day that passes could result in a spike in his ammonia levels that could cause permanent brain damage. So time is of the essence in this disease, and so they made every effort to work quickly to develop this for KJ. So how is KJ doing since he had his therapy? His disease has improved. He is able to tolerate more protein in his diet now, but he still takes medication, but less medication than he did prior to the treatment. So it's been helping to keep his ammonia under control. He has had a few infections since his treatment, and his body handled them very well, whereas normally in these patients, their ammonia levels would spike when they have an infection. And so that also looks very promising. He is being carefully monitored at CHOP, and to date, he hasn't had any serious side effects. But longer-term follow-up will be needed to really fully evaluate the benefit of his therapy, and he will have to be carefully monitored for the rest of his life. So how was the treatment funded? I think this is really an important point to highlight. So the National Institutes of Health was really essential in funding this work. Most of the gene editing treatments that are being developed for rare diseases are being developed by researchers at universities like Dr. Bauer's group. The hope is that this work will pave the way for creating treatments for other rare diseases in the future. So not just for SDS or CPS1, but for many genetic diseases. And I think it's also important to keep in mind that as the field gets better and better at doing this type of therapy, it can be applied to many, many more patients. And that is the hope. Platforms are being set up to make this feasible in places like CHOP and at places like Boston Children's Hospital. So the infrastructure is being set up to do that for many rare diseases. So I think as highlighted in the session this morning, gene editing certainly holds promise, not for just CPS1 deficiency, of course, but many rare diseases that are caused by these misspellings in the DNA. And all of those, or many of those, could be fixed with this type of treatment. Many of these rare diseases also don't have any approved therapies. And so it's really hoped that this will be applied across many diseases. And I think the KJ story really shows that it was possible to rapidly make this personalized therapy. From the time he was diagnosed to the time he was treated was about 7 months. And so I think this exemplifies how this could happen in the future. Dr. Ahrens-Nicklas stated that years and years of progress in gene editing and collaboration between researchers and clinicians really made this moment possible. And while KJ is just one patient, we hope he is the first of many to benefit from a methodology that can be scaled to fit an individual patient's needs. So what is the status of gene therapy for blood disorders? As mentioned earlier in Dr. Bauer's work, there are gene therapy approaches, including gene editing, approved for sickle cell disease and beta thalassemia. And as highlighted in those earlier talks, these treatments take the patient's own blood stem cells and modify them in the laboratory. And then these modified cells are infused back into the patient's body through a bone marrow transplant. So as Eszter highlighted, these are known as ex vivo approaches. Importantly, to help those cells grow back in the patient's body, strong chemotherapy is required. And this can be harmful and toxic, but there's a lot of interest and excitement about developing less toxic chemotherapy approaches that can be used in these settings. In vivo gene editing is something that there has been a lot of interest in. And so the idea here, as Eszter mentioned also, instead of fixing the gene in the laboratory and doing a bone marrow transplant, those genome editing tools would be delivered into the body. To do this, the lipid nanoparticles would be used with special modifications that can actually target them specifically to blood stem cells. And this work has been developed by Dr. Stefano Ravella at the Children's Hospital of Philadelphia and his team at CHOP. And they've done this type of project in a mouse model and demonstrated that they can target those stem cells and they can use genome editing. And it does work in this preclinical model. The advantage of this type of approach may be that chemotherapy may not be needed. And so I think this really highlights that there's a lot of active research ongoing to try to develop novel approaches that could allow for an in vivo gene editing approach. So how do all of these approaches impact SDS and other bone marrow failure syndromes? So as I highlighted, less harmful conditioning regimens are being investigated that would be important for making bone marrow transplants more feasible for SDS, as well as other bone marrow failure syndromes. As you saw in Dr. Bauer's work, gene editing approaches are being designed for SDS specifically. And in vivo gene editing may provide a tool for correcting the gene with less toxic conditioning. These treatment approaches can be applied to other rare diseases, and the platforms are being set up that can be used for SDS. I think most importantly, I think that it's an exciting time because this is such an active area of research. And, once in vivo approaches are developed for other blood disorders, and these reach the clinic, this also helps pave the way for SDS. The SDS field can learn and benefit from these approaches, and further, it paves the pathway forward in terms of regulatory aspects as well. So I just want to acknowledge Dr. Ahrens-Nicholas and Dr. Musunuru and their teams for all of this work that's led to KJ's story. And here at CHOP, of course, we always want to thank KJ and his family for their willingness to let CHOP share their story. And the blood stem cell approaches that I highlighted are being developed by Dr. Stefano Ravella and his team at CHOP. So thank you very much.
Thank you, Dr. Sabatino. If there are any questions from the audience, please type them into the Q&A or into the chat window. We got one question so far, and I think it relates to baby KJ specifically, whether he was treated with, I guess, conventional treatment prior to gene therapy.
So, well, first I will say I'm not KJ's clinician. And so I don't know all the details of his treatment prior to the genome editing therapy. But I know that they use standard of care to care for him until he was able to receive the gene editing therapeutic. So they used, but I don't know specifically if they use lactulose. So thanks for the question.
And I have a question too. When you think, or when we think about like side effects, like we know with the conventional gene therapy for blood disorders, it comes with a transplant and conditioning. So there's a lot of side effects and toxicity expected. What types of side effects and toxicities would be expected for an in vivo gene therapy? Like, is it really just people go in, receive the treatment, and then walk out, go home, or are they expected to experience some immediate side effects?
Well, I think in KJ's case, they did not observe any significant side effects of concern. The lipid nanoparticles they used targeted the liver very efficiently. So then as we think forward about applying these approaches to target, for example, the bone marrow, I think less is known at this point in time. And the studies that have been performed are in animal models. So I think it's a little less clear and it's a little more complicated, but I know that the investigators, such as including Stefano Ravella and his group, are really interested in understanding that in more detail so that it can be applied to a clinical setting.
And any last minute questions, anybody? Type them into the QA. If people think of more questions later, please email it to me and we'll discuss it with Dr. Sabatino and gather some answers for you. But it looks like for now, we're good on questions. So thank you. Thank you so much, Dr. Sabatino, for sharing these insights with us.
Thank you so much for having me.
Next, it's almost time for the lunch break, but we skipped over Dr. Elefanti's work. So we're gonna go back to that. That's also a recording because the research group is in Australia, which is a different time zone altogether. And the group has been also at the gene therapy conferences in Spain this past week. But he graciously recorded a presentation for us. So this is the work of Professor Andrew Elefanti and Dr. Elizabeth Ng. And the title is Induced Hematopoietic Stem Cells multilineage engrafting hematopoietic stem cells from human-induced pluripotent stem cells. So that's a mouthful. I just wanted to summarize quickly what's different from this work compared to Dr. Bauer's work. So Dr. Bauer's work proposes to mobilize the hematopoietic stem cells from patients, take them out through apheresis, and then edit them and then put them back into the patient. Dr. Elefanti's work is a little different. So instead of taking hematopoietic stem cells from patients, they can use just regular blood or skin or other cells and reprogram them to become induced pluripotent stem cells. So those are iPSCs, which then with further treatment can be made into any type of cell in the body. It could be made into neurons or heart cells or muscle cells. And more recently, the group has developed a method to reprogram iPSCs into engraftable hematopoietic stem cells. So the difference from this type of approach compared to Dr. Bauer's approach is that they could take any type of cell from a patient earlier in life, possibly, and then if and when treatment is needed, those cells could be reprogrammed to become hematopoietic stem cells, could be fixed with gene therapy, could be expanded, so they could make more of it than they could get from getting the hematopoietic stem cells from the patients directly. And so there could be some treatment benefits and other opportunities in that type of approach. The gene therapy or the gene editing part itself might be exactly the same. It's more about what types of cells are used to do the gene therapy on. So we'll play the video now. If people are starving, you're also welcome to take your lunch break now, and we will be back at 12:30 Eastern Standard Time.
Good morning. My name is Andrew Elefanti, and I work at the Murdoch Children's Research Institute in Melbourne, Australia. I'd like to thank the organisers for allowing me to have the opportunity to tell you a little bit about our work with induced pluripotent stem cell derived hematopoietic stem cells. My disclosures are that I am a consultant to Retro Biosciences. The story I'm going to tell you has really been a long time in its generation. It's a long journey that's involved Ed Stanley, Elizabeth Ng, and myself really over 25 years. We've brought together our complementary skills in cell developmental and molecular biology and our shared desire to generate blood cells from pluripotent stem cells. The way we've envisaged this as working is taking iPS cells derived from patients, differentiating these into induced hematopoietic stem cells, which can be used for a variety of applications, as I've shown on this slide, including cell therapies where replacement cells for malignant and non-malignant hematological disease can be provided. We can also use this system to model blood diseases to both study the pathogenesis and improve treatment, for example, by screening for drugs or screening for gene therapy vectors. And as well as that, there's the ability to make blood cell lineages from the differentiated hematopoietic stem cells, which are therapeutic in their own right. Focusing on the cell therapy aspect, the idea is really that reprogramming patient cells to iPS cells can generate autologous induced hematopoietic stem cells that circumvent problems of donor shortage and graft versus host disease. And the way this works is as shown here. The patient cells are reprogrammed into iPS cells. And at this stage, patients who have underlying genetic mutations, for example, like those patients with Shwachman-Diamond Syndrome, can have these corrected at the iPS cell stage. These cells can then be differentiated into induced hematopoietic stem cells, which then can be a source of an autologous cell therapy. The range of diseases such as bone marrow failure that can be treated like this it is really a very large number. And to give you an idea of the scale, approximately 90,000 hematopoietic stem cell transplantations are performed per year. In bone marrow failures, the stem cell therapy replaces the blood system of patients who have got marrow failure, curing the cytopenias and preventing progressions to myelodysplasia and leukemia. And conversely, patients who have leukemias or lymphomas, stem cell therapy regenerates a blood system, rescuing the patient following chemotherapy. So late last year, our laboratory published a paper describing the generation of long-term engrafting multilineage hematopoietic stem cells differentiated from human pluripotent stem cells. This met with a lot of interest and enthusiasm around the world, and we are fortunate to be graced with the front cover of the prominent journal Nature Biotechnology in August of this year. So how does this actually work? How did we actually make induced hematopoietic stem cells from iPS cells? Well, we essentially tried to recapitulate the process of human blood development in a dish. We take induced pluripotent stem cells, we lift these off the plate and turn them into little aggregates of cells, which we call embryoid bodies. These are shown here in red because we've colored them with a fluorochrome, which enables us to detect them more readily after transplantation. Over a period of days, in response to various growth factors and small molecules that we introduce into the medium, these embryoid body structures grow. And as you can see, from day 7 onwards, they start to develop these protuberances on the surface, which represent the forming blood cells emerging from endothelial precursors. These increase in size over the ensuing days and are shed into the medium as blood cells. So the blood cells at this stage can be cryopreserved, and then when we wish, they can be thawed and transplanted intravenously. And in this case, we are using immune deficient mice as a model system. And this in a way replicates the normal process whereby hematopoietic stem cells are transfused into patients. We can monitor the mice looking at the presence and engraftment of human cells into the peripheral blood after 12 and 16 weeks, and we can harvest the hematopoietic tissues after about 5 months. And in this example here, we can show the presence of human cells in the bone marrow, in the spleen, and in the thymus by flow cytometry. And if we're using the cell line, which is colored red, of course, we can see these cells as red cells in the bone marrow by fluorescence. So this gives us an outline of a sort of a vision towards the way we can see developing a patient-specific therapy, where there's primary samples taken from the patient, reprogrammed to iPS cells, any mutations present are corrected, and then these cells are dissociated and differentiated in bioreactors such as this vertical wheel bioreactor that we've got here. The blood cells are generated, they are then harvested off and can be transplanted as a corrected CD34 positive autologous stem cell product back to the patient. We're very excited to announce that earlier this year we joined forces with the US biotech company Retro Biosciences, and we're working with them to generate a personalized patient-specific therapy that can be used to treat children and adults with a range of bone disease, bone and bone marrow failure and other hematopoietic diseases. So we are hoping to map the pathway towards a first-in-human clinical trial of an autologous iPS-derived hematopoietic stem cell therapy for patients with diseases such as bone marrow failures and leukemias. And we want to develop a manufacturing pipeline for the provision of the next generation of patient-specific therapies to revolutionize the whole process of hematopoietic stem cell transplants. We are very mindful and sort of keen to continue our work with patient advocacy groups such as the Shwachman-Diamond Syndrome Alliance, because we really recognize the critical role that these play in supporting patients and their families, and it's vital to ensure that their voice is heard when new therapies are being developed. So with respect to Shwachman-Diamond syndrome, you know, where are we at now? Well, we are fortunate and grateful to be supplied by iPS cell lines from a patient, female patient with Shwachman-Diamond syndrome, which were generated by the Coriell Institute for Medical Research. We took these cells and we corrected one of the mutant alleles to generate a heterozygous, therefore clinically normal cell line. And in our preliminary investigation so far, we've been able to take both the patient and the corrected cells. We can differentiate them via the protocol that I showed you early on in vitro, and we can demonstrate that they, both the patient line and the gene-corrected line, make CD34 endothelial cells with a similar efficiency. And when we differentiate these through the blood cells, they have a similar spectrum of expression of hematopoietic stem cell markers on the cell surface of these cells. The actual name of these markers doesn't matter, but what I'm really wanting to show you here is that the patient line and 3 corrected clones are all essentially behave very similarly. And similarly, they, the patient and the corrected clones have a similar yield in terms of the blood cells that are generated. So where do we go to next? Well, we want to further differentiate the stem cells in vitro to look at the way that erythroid and myeloid cells develop, to study how blood cell development occurs in patients with the disease. This will enable us to also set up screens for beneficial drugs or potentially test therapeutic gene therapy vectors. We can also look to functionally evaluate the patient and the gene-corrected hematopoietic stem cells in immunocompromised mice, a bit like I've shown you earlier, where the idea is we could take hematopoietic cells both from the patient and from the corrected lines, and we can transplant into the mice. And in particular, we'd like to see whether the corrected cells can actually outcompete and replace the patient cells. So this schematic here shows an overall picture of how everything might work. We've got patients who have Shwachman-Diamond syndrome. You know, we're aware that you know, they have a predisposition to myelodysplasia and leukemia, and this increased chance of blood diseases increases with age. So, you know, what we're proposing might be a therapy for this will be if we make iPS cells from these patients, we can correct these in vitro, we can direct these to differentiate into making autologous hematopoietic stem cells, and then we can transplant these back into the patients. We may be able to prevent the leukemia and improve the patient outcome. So in summary, I've shown you in these last few minutes that reprogramming patient cells to iPS cells enables differentiation to autologous induced hematopoietic stem cells. These iHSCs allow us to study diseases, to screen drugs or gene therapy vectors to improve treatments of blood disorders. The gene-corrected iPS cells allow the development of a new autologous hematopoietic stem cell therapy, which would not have problems of donor shortages or graft versus host disease, which can be causes of difficulties with allogeneic transplants. And these therapies, we hope, may reduce the risk of progression of disorders like Shwachman-Diamond to myelodysplasia or to leukemia. And I'd just like to thank the many people in our laboratories who've been involved in this project, and of course the mice that have helped us to work out the protocols to make the induced hematopoietic stem cells. And I'd like to acknowledge our collaborators and funders, and I thank you for your time.
Hello, everybody. Welcome back. We have some really interesting talks lined up for you, so let's jump right in. So the afternoon session is all focused on how we as patients can make a difference. And to further that idea, we also need to understand how drug development works. So our first speaker for the afternoon is Kendall Davis. But I wanted to position this topic a little more. We will basically, we talked about translation in the morning session already quite a bit. We talked about the normal understanding of the word translation, which is translation into different languages. We want these talks to be accessible to patients anywhere in the world. Then there's the translation of translating the information encoded in the DNA into proteins by ribosomes. That's also called translation. And then the third concept of translation that's highly relevant here is how can we apply what we learn in a lab setting to actual patients? So how do we take our learnings and methods developed in a lab and make it so that it can benefit patients? So our first talk this afternoon will focus on how clinical trials work. That is the step of taking knowledge into patients. In the graphic here, it's depicting like a petri dish and mice. So a lot of learnings coming out of there, and how can we convince the FDA and other regulatory agencies that it's safe and effective and worthy of trying in patients. And then the second talk in the afternoon will focus on another aspect of the challenges around rare disease, is that even if the science and the regulatory challenges are worked out, there's still a question of the economics, how to make it work financially in small populations. So that will be our second talk in the afternoon. So with that, it is my pleasure to introduce Kendall Davis. She's currently head of clinical engagement at Sano Genetics, but she has a long history of experience with clinical trials. And she's also worked with Team Telomere quite intensively, which is another bone marrow failure disorder, and a great patient advocacy group that focuses on patients with telomere biology disorders. The title of the talk is a little open-ended, but basically we'll learn about the phases of therapy development and pathways to success. Welcome, Kendall.
Wonderful. Thank you so much for having me. I'm so excited to be here. Like said, my name is Kendall Davis, and I'm the head of clinical engagement at Sano Genetics. I have previously worked across all phases of clinical drug development, all very specifically within the context of rare disease. So hopefully I'll be able to provide a higher level overview and get into a little bit of the granularity with regard to how clinical research works, and more specifically how patient advocacy organizations and patients such as yourselves and community members can be involved in this process to make sure that we're creating therapies that are meaningful to the community, but also very accessible. Quick disclaimer, the information I'm going to share today reflects my own personal and professional experience and is not necessarily the views of any particular sponsor, organization, or regulator. So before we really dive into specifically clinical trials, I want to set the stage for what standard clinical research looks like. So clinical trials are a piece of this larger context of rare disease, or I'm sorry, standard clinical research. So before we get into clinical trials, we have the preclinical phase. So this first phase here. And so essentially this is the phase. So before testing in humans, researchers study a therapy in a lab or in animal models to understand not only how it works, but how it might be processed in the body and whether it appears to be safe enough to move forward into clinical research. Once this piece has been concluded, either sponsors or academic institutions, whoever is doing this research, will file a IND or an investigational new drug application. And so again, if that preclinical research looks really promising, this application will get submitted to regulators like the FDA to request permission to begin testing the therapy in humans. There has to be an approved IND before clinical research can happen. And again, this is really to make sure that everything that is going to be studied in humans is safe. So once the IND has been approved, then you can start the clinical phase of this process, so clinical trials. And there are 3 phases of clinical trials that we'll get into in a minute, but they're phases 1 through 3, and they evaluate the safety, the dosing, and the efficacy of a potential therapy in human beings. And then once we hopefully will see a very effective and positive result from clinical trials. So once clinical trials, all phases have been complete, the sponsor will submit an NDA or a BLA, so a new drug application or a biologics license application, depending on the type of therapy, to regulators. Within this application, they're going to provide all of the data needed to demonstrate that the therapy is safe, effective, and can be consistently manufactured. So this is really important. You have to be able to show that not only do you have a therapy that's safe, effective, it looks really great, but also that it can be manufactured in a way that's going to make it accessible to patients. After this submission has happened, it will go through regulatory review. So again, this could be the FDA here in the United States or the EMA in Europe or different regulatory agencies around the world. And the regulators will look very, very closely at all of that data submitted. They often have consulting advisories or advisory committees that will look at very specific aspects of applications to decide whether they think that it meets the standard for approval. If all goes well, a drug will be approved and it'll move into the approval phase. And if there's evidence to support the formal approval and all of the scientific conditions or everything has been met scientifically, it will allow the drug to start being marketed. And so once a drug is able to be marketed, then it will become commercially available to patients. After this happens, usually there is a requirement for a post-market or a phase 4 study that will continue to look at the safety and efficacy of a drug long-term. So once it's been approved, there still is an option for continued evaluation to make sure that safety standards are continuing to be met. So what is a clinical trial? We talked a little bit about this, but the basics are that clinical trials are research studies where volunteers, people like you or families you might know, help to answer key questions about a potential new therapy as it relates to being safe and effective. Trials are how medicines really come to life, and they test not only if something works, but they're looking at how it works, for whom it works, and how safely it works. It's really important to note that trials also explore new ways to use existing treatments. So for example, they might look at changing the dose, the delivery method, or the population of people that a drug is approved for. And they are always looking to try to make them more accessible and effective. So who's doing a clinical trial? There's so many components to clinical research, and there's a lot of different players in this game. This really illustrates kind of a high-level overview of the major players in clinical research. You have the regulators like the FDA or the EMA who set the standards for the research, the life sciences companies or potentially academic institutions that design and fund the research, CROs or contract research organizations that handle all of the logistics and ensure the compliance, the physicians and specialty centers who are actually running the trials at their sites and providing all the care for participants. And then at the heart of it all, you have the participants themselves. And every decision in this process really must prioritize the safety, informed consent, and well-being of participants. Those are the standards that have to be met for any research to happen. So in clinical research in general, there are some pretty standard objectives. So the goal of every trial is to generate enough credible evidence to support market authorization or regulatory approval. So everything we're doing is to get this body of data that's going to allow a drug to come to market. To get there, trials need to demonstrate that they're both safe and effective. They're basically two sides of the same coin. And the evidence that is presented must be adequate and well controlled regardless of the disease type. So there are some pretty specific objectives and standards that have to be met. So we talked a little bit about the phases of clinical research in general, and when we're talking about clinical trials, they tend to happen again in 4 distinct phases. In phase 1 trials, this is often when a therapy is being tested in humans for the very first time. It's focused primarily on safety and determining the right dose range and identifying any potential side effects. This is usually done in a smaller group of healthy volunteers, but because rare disease trials are a little bit different, sometimes we do see phase 1 trials actually happening with rare disease individuals themselves. Phase 2 studies are trials that explore whether a treatment works as intended and they also continue to evaluate the safety. They typically involve a larger number of people and with a very targeted condition in mind. When you get into phase 3 trials, these are usually considered the most important or the pivotal trials within clinical research. They're usually a little bit larger. They're still well-controlled studies that confirm the treatment's effectiveness. They continue to monitor any side effects. And again, they're looking at a little bit larger of a population of people. And they're comparing this therapeutic to either existing standards of care or something else to support that regulatory approval. So usually phase 3 trials are considered some of the most important trials. And then again, if everything looks really good, you might have a phase 4 study, which is that post-market study that looks at the long-term safety and efficacy in a broader population of people. I do just want to point out that in rare disease or in any area where you just don't have as many individuals, things get quite a bit more complex. And regulators have started to recognize this, and they recognize that when patient populations are smaller or more nuanced, the standard design may not work. So you might see trial designs vary a little bit. They might involve fewer patients. They might combine phases of study. So you may see something that says it's a phase 1, 2, and so it's combining different phases of development to look at things in tandem, as well as, like I said, it might start with actually patients themselves instead of healthy volunteers. So there are ways that we are trying to evaluate how we can create clinical research in these rare disease populations to, again, bring therapies to market as soon as possible. But even within this flexibility, the requirements of studies being rigorous and well-controlled, that doesn't change. So the balance, it becomes a balancing act of scientific rigor and practical feasibility when we're talking about rare disease. And so again, we'll talk about later the ways that patients and patient advocacy organizations can kind of help this process so that, again, we're setting ourselves up to have the best clinical trials possible, given the populations that we're looking to research. There's a ton of terms within the clinical research atmosphere and in clinical trials, but I did want to point out just a couple that are really important to understand and come up quite a bit. And these two terms are clinical endpoints and surrogate endpoints. So these are terms that you're going to always hear in the context of clinical trials. So I want to kind of provide a little bit of a differentiation between the two. So a clinical endpoint is these are measurements. So these are the things within a clinical trial that are going to be measured. Clinical endpoints are very direct. They measure whether a patient feels better, functions better, or lives longer. So it's looking for a very clinical output of a potential trial. Surrogate endpoints are usually a little bit more indirect. They're like biomarkers or lab values that might predict improvement. So for example, if we think about like a heart disease study, survival or exercise tolerance would be a clinical endpoint, while troponin levels might be a surrogate endpoint. So they might be looking at a lab value as a surrogate endpoint, but actual survival or ability to tolerate exercise as that primary clinical endpoint. So how are clinical trials designed and how are they controlled? So clinical trials, the design of clinical trials is an incredibly intense and complex process. We have seen this start to include patient organizations and patient voices much earlier because we have seen as an industry that the sooner we can get insight into a protocol or a design, the better likelihood we have of creating a clinical trial that is going to be accessible and meaningful for patients. it's important to understand that every trial follows a very specific protocol. And a protocol is essentially a detailed blueprint that describes who can participate, how the study will be conducted, what data is collected, and what success of the trial is going to look like. Trials rely very heavily on controls to ensure that the results are actually meaningful and we're able to see them. So we usually compare an investigational treatment, so something being studied in clinical trials, to either another treatment, no treatment at all, or a synthetic control arm that might be modeled after real world data. So sometimes in studies, we can take bodies of data like a natural history study and study what a therapeutic looks like compared to the natural progression of a disease. Without comparison, we can't know if an observed benefit is real or coincidental. So all research has to follow a protocol and it has to be compared to something. So it has to be compared to a control. Within clinical research, bias is it's a huge issue. So whenever evidence for anything is concerned, bias is considered the enemy and it's everywhere. So bias is something that can lead us to believe that something is or isn't working believe that something is or isn't true despite evidence on the contrary. So trials have to be designed with measures to intentionally eliminate potential biases. There are three very common approaches for eliminating bias within clinical trials. They are randomization, blinding, and placebo controls. One thing that they have in common is that they involve not knowing on the part of the people involved in the experiment, which can feel very frustrating to the participants, but I can tell you that it's critically important to remove biases and hopefully bring drugs to market faster. Randomization eliminates selection bias. So a researcher in a space might unconsciously group some people with similar characteristics into one group or another. And so selection bias is the practice of randomly assigning participants to either a treatment group or a control group. And it's important to note that randomization might not always be 50-50. So sometimes there might be less participants getting a placebo as opposed to the therapy. And it's all going to depend on what's appropriate for the evidence that needs to be generated. Blinding eliminates something called confirmation bias, which is when a person or researcher or someone analyzing the data might unconsciously interpret information in a biased way. So the way that we blind, again, is by not knowing who's getting a treatment and who might be in the control group. And to keep studies unbiased, we also use randomization, which again is assigning participants to group by chance, so that neither the investigator or the patient will know if they're receiving the therapy. Placebo-controlled studies often spark a lot of debate, specifically in rare disease or less common conditions. But they remain critically important in many cases. They provide the clearest evidence as to whether a new therapy truly works, which ultimately is going to speed up regulatory approval and access to everyone. Placebos keep participants and possible physicians from knowing who's getting a treatment and who's not. Often people think of placebo as like a sugar pill, but it essentially would look exactly like however the therapy is being delivered. So it could be an injection or something else. It can feel really disheartening to imagine being in a clinical trial and getting a placebo. And this is something that there's a lot of questions around. And so people who are designing studies think very critically about whether or not to introduce placebo controls into a trial. But it's so important to remember that this really is the fastest way to get through regulatory review and show whether or not a therapy is working and whether or not it should become available to the patient population. So who can participate in trials? This is also a really big question. Every study is going to have defined inclusion and exclusion criteria. So the medical or demographic traits that determine eligibility. These criteria are designed to keep people safe and ensure that the data is reliable, again, to make sure that we're moving things as quickly as possible. That said, over-restrictive criteria can sometimes make clinical research inaccessible, and many advocacy groups today are actually working with sponsors to create realistic inclusion and exclusion criteria to reflect the real world populations. So this is something that your organization or you might be able to participate in doing some of the protocol reviewing and thinking about the criteria, the inclusion and exclusion, and how we can actually map it to what the population looks like. So taking part in clinical trials, this is such an important decision that individuals make. So it's really worth emphasizing that this is a very, very personal decision, and there's a lot that should go into the decision making around participating in a trial. Deciding whether or not you want to is deeply personal, and there should never be any type of pressure to participate in a study. Clinical trials can require significant time and travel, and it often will affect an entire family. So one person participating can have like a pretty incredible impact on loved ones or a family ecosystem. So thinking about what a clinical trial is going to look like within the context of your life is very important. For some people, it's worth it and they want to participate, and for others, it may not be. So again, it's really important to think about everything that's going to be asked of you when participating in a trial and determine whether or not that's going to be something that can fit into your life. Ask a ton of questions. You should always feel like if you're being talked to about a clinical trial that you can ask everything you'd want to know about what participation is going to look like so that you can make as much or as informed of a decision as you can. Again, reasons to participate. These are very personal. There's a lot of great reasons to participate in clinical trials, but definitely make sure, again, you're taking into consideration all the aspects of it and whether or not this is going to be the best option for you at this time. So one of my favorite things and something I've spent a lot of time working on throughout the past many years, is how the community, how patient communities can really support clinical research. This is an incredible graphic that kind of walks through all the phases of research and how patient communities have become more involved. And so it's important to note that organizations are playing an essential role in making trials more successful, from shaping study design to helping sponsors reach diverse populations and even supporting families through the process. So collaborations between industry, clinicians, and advocacy groups is what we're seeing as transforming trials from more scientific exercises into true community efforts. So I could spend hours just talking about this slide alone, but it's important to know there are a lot of different ways that you can engage as a community in clinical research. And as an organization, ways that you can not only support sponsors or academic institutions who are designing and running trials, but also the community in participating in clinical research. The Clinical Trial Transformation Initiative did this incredible exercise where they looked at patient group involvement across the research continuum. And it was pretty amazing to see just how much impact patient organizations can have throughout the entire life cycle of drug development. I would encourage you to get to know different ways that you might personally be able to participate or even support your organization in making sure that we're optimizing clinical research, not just for the researchers, but also for the patient community. Want to talk a little bit about some of the programs that exist to make therapies more accessible. These are things that you might hear about a lot when it comes to clinical research, but aren't as well known. So shifting a little bit, these programs are called managed access or compassionate use programs. So sometimes there are situations where patients can't join a clinical trial, but still need access to an investigational therapy. And so there are a few mechanisms that make this possible. They're here, and I'm going to walk through each on their own. So expanded access programs or compassionate use are programs in the United States which allow for the use of an unapproved drug outside of a trial when no other option exists. So this is when if there's a drug being studied in clinical trials, but it's not approved by regulators yet, physicians could decide that a patient should have access to this, and they can request access through either expanded access or compassionate use programs. There's quite a bit that goes into this, but it is important to know that this is an option and it is something you could speak to your physician about. The Right to Try Act in the United States provides an alternative path for certain late-stage investigational drugs. So again, this is just another way that individuals could get access to drugs where they might not have been able to participate in a clinical trial, or there may not have been another access point. There are some criteria listed here for the investigational treatment itself, as well as criteria for the patient. But it is important to know that the Right to Try Act does exist. And again, it's something you could talk to your physician about, or even your advocacy organization, to make sure you understand if this is ever something that would be applicable to your situation. The named patient programs are programs throughout Europe where a physician can request a therapy directly for an individual patient without regulatory oversight. So this is similar to our expanded access or compassionate use programs, but these are different, and there are different criteria depending on the country in Europe that have these programs. These pathways really balance compassion with safety. So they're looking to make therapies available for the people that need them, while also making sure that we're doing so in the safest way possible. They want to ensure access that doesn't compromise ongoing research. So these are meant to, again, create that balance between ongoing research and therapies that are in development, and the ability for patients who really need them to have access. So what happens beyond drug approval? So once a therapy is taken through clinical research, and we really hope that it passes or it meets its endpoints in phase 3 and it gets approved. But approval is not the end of this process. It's kind of the beginning of a whole new phase. So once a therapy is approved, companies will start working with payers and healthcare systems to make it available. Patient organizations can often step in here again, helping with reimbursement, advocacy, education, and copay assistance programs. So this is a really cool way where patient organizations can be a part of the conversations with payers or health systems that are going to ultimately have to approve these therapies, to make sure that the need for them is understood, and the impact that having access to these drugs would have on the overall health of an individual. For patients, access means a lot more than approval. So it means that the treatment can actually reach them. It's affordable, it's equitable, and it's sustainable. So like I said, there are a lot of ways that organizations can help sponsors or academic institutions who's ever trying to make drugs accessible, have those conversations with the people who are going to be kind of in charge of the access point, to make sure it's understood why individuals need them, what it's going to do to their overall quality of life and healthcare, and why it's so important that they're as accessible as possible. So I would like to say thank you so much for having me here. Hopefully this was a nice high-level overview of what clinical research looks like, specifically clinical trials, and some of the ways that patient advocacy organizations and patients such as yourselves can really be a part of this process. I want to just point out that every approved therapy that we have today exists because someone, a patient, a family, an organization, a group, chose to participate in research. So whether you're a clinician, a community leader, an advocate, all of you play such a critical role in this journey. And it is so important for all of you to understand how amazing it is that you are even considering either being a part of this system or educating about it, because it is critical to bringing therapies to rare disease or smaller groups of people as fast as possible. It means the world to me that people consider clinical research, and that we are seeing more involvement from patient organizations. So for any of you involved in this process, from the clinic or sponsor academic side, thank you so much for all you do. And for anybody who might consider participating in research, please know that it's not lost on us as an industry how much we're asking of you in clinical trials, and how just imperative it is, and how grateful we are for your even consideration and involvement. Thank you so much.
Thank you, Kendall. That was a wonderful overview, and I especially appreciate how it's becoming really clear that patients and patient communities are an actual partner, not just a guinea pig, so to say, but we really have to play an active role if we want to see therapies developed and benefit us. So thank you for highlighting that. To the audience, please type your questions into the chat window. We have one question so far, and it's related to the informed consent process, or ASCENT. For our disease and many other rare diseases, there are a lot of children involved, and how do we make sure that we're responsible in enrolling them and that they have a say in it? So can you explain the difference between informed consent and ASCENT?
Yeah, sure. So I will do my best. So informed consent is the process of essentially explaining everything that's going to happen within a clinical trial, everything that's going to be asked of you and that you would be doing, to make sure that you understand all aspects of a protocol and a trial before you decide whether or not you want to participate. Informed consent it's a very intense process. Usually informed consent documents are very long because they're meant to provide you with all of the information that you could possibly need, hopefully, and it should be done with either a physician or a coordinator there to answer any questions. And it's something that I would encourage, if you are going through an informed consent, make sure you really are asking all of your questions. It can be a very intimidating process, I've heard from individuals, because you're reading a lot of information that might not make a ton of sense to you when you're first reading it. So definitely ask as many questions as you can, because that process is meant to provide you with a baseline to determine whether or not you want to participate. When we're talking about trials with children or any pediatric study, we have to consider ASCENT. And so this is basically the informed consent that a family will go through to determine whether or not their child, who isn't an adult, should be participating as well. And so when we're talking about ASCENT, this is, again, there's a lot of nuance that comes in here, and a lot of it comes down to, as a family and as a parent or a caregiver or whatever the situation is, like how you're determining the level of involvement you want the participant to have in this process. There has been a significant amount of work to try to make assenting a lot more accessible to the people and the individuals that are going to actually go through this, so the kiddos or younger people, to make this a little bit easier. But just to say, it's still incredibly intense, and we're asking usually parents to make decisions about whether or not their child should participate in a clinical trial. So again, I would absolutely suggest asking all the questions, making sure that the individual who's going to participate to the best of their ability can ask questions or understands. And there are some incredible resources I'm happy to share after this that kind of go through this a little bit more. But my biggest takeaway here would be to use the time in either of these informed consent or ASCENT processes. Take as much time as you need to have those conversations with the coordinator. It may feel like a rushed process because we know that a lot of times medical professionals they're very busy, but this is your time, and you have every right to take as long as you need and ask as many questions until you feel comfortable making a decision about participation.
Thank you. Thank you. And we also just got some great feedback for you, Kendall, people appreciating the explanation of compassionate use, because we talked about those terms in the morning session, so it's very timely. And I don't see any other questions at the moment. So thank you again, Kendall, for walking us through these very complex topics to try to wrap our heads around them. And I'll monitor the questions and forward them any more questions if they come along.
Please do.
Yeah, thank you. Now moving on to the next one. Oh, so I just prepared 1 or 2 slides because we talked about clinical trials. Where do we even find them, right? Like sometimes we will hear about them from our favorite doctors, because oftentimes if they are running a trial that's relevant to us or our kids, they will tell us about it. But there are likely to be other trials out there that those physicians are not familiar with or not involved with, but you might still want to explore and ask your physicians about them. So there is a database that the US government maintains. It's called ClinicalTrials.gov, where investigators can submit their trial to be listed. So I'm not going to go through it in great detail, but it's literally just a database. It's not an endorsement by the government of said trials. And so if you go to ClinicalTrials.gov, and they recently overhauled it, trying to make it more user-friendly. And there are secondary websites, commercial websites that take this information and repackage it to try to make it even more user-friendly. But in essence, there is, if you go to ClinicalTrials.gov, there's this search window with multiple fields on the top. So it's depicted on the top left of my slide. And so you could type in, for example, Shwachman-Diamond Syndrome. You click search, and then you get a list of all the clinical trials that include that disease or gene or intervention or therapy as a keyword. And then you can click on any of them to learn more. The example I'm showing here is actually our SDS GPS program. So that is also considered a clinical trial, but it's not interventional. So we are not administering any drugs. We're collecting data to learn more about the disease. So it's sort of like a registry, and registries can be listed on ClinicalTrials.gov as well. If you do this search yourself, I think currently there are 2 or 3 pages worth of search results. Another one is a clinical trial by X4 Pharma. And if you are an adult with neutropenia, with SDS, of course, and neutropenia, and have experienced, I think, like 2 infections within the past 12 months, look into that trial more and discuss with your doctor whether it might be right for you. It will explain all the age limits, inclusion criteria, exclusion criteria, as we learned about just a few minutes ago. So with that, we can move on to the next talk. And again, to frame it, going back to this idea of translation. So now that we covered the steps and the strategies involved in taking some findings from the lab to actually benefit patients, another aspect of that is the economics. How can this be made to work, given that we are dealing with very small patient populations, and so the commercial benefit from just the market is just not there in the same way as a blockbuster drug. So we have to think creatively how to overcome those barriers. And we have a speaker to walk us through those ideas. So I'd like to welcome Joe Katakowski, who is Director of Research at the RTW Foundation. And as mentioned, he will walk us through the challenges and opportunities for rare disease research and development from a regulatory and economic perspective.
All right. Thank you, Eszter, for the introduction. I'm going to just share my slides. Hopefully you can see them. Get them onto, there we go. Okay, you should see my title slide now. And can you hear me?
Yes, we can see the slide and hear you well. Thank you.
Great. Okay, so I'm Joe Katakowski. I'm going to tell you a little bit about the, well, it's a combination of both. Regulatory sort of informs the economic challenges and the manufacturing challenges facing cell and gene therapies and genetic medicines in general. which are usually the most productive paths forward for genetically defined disorders amongst the rare diseases. A little bit about the foundation and what we do. My background is in preclinical research and development. I work at the RTW Foundation and provide advice and support to other rare disease foundations that are pursuing therapeutic research efforts. So the information that I'm conveying today comes from my experiences in interfacing with multiple different ultra-rare disease foundations at varying different levels of drug development efforts. The foundation I work for provides this advice for free through the Rare Disease Advising Program. We have a very small grant cycle as well that we help to catalyze preclinical research. But the majority of our role is in helping rare disease foundations find their research partners or the best research partner for their modality, and helping to get these research foundations to a position of savviness to where they are really the center of the universe for their drug development efforts. And that's sort of the goal that we seek to do. You can learn more about our organization at the website there. I'm going to do a little bit of discussion on definitions around rare disease and why they're neglected. And this might not come as a surprise to anyone on this call if you've attended conferences sponsored by NORD or Global Genes or Every Life. This will make perfect sense to you, but for others, it can be an illuminating conversation. So rare disease is a spectrum. Its definition depends on your geographic location, essentially. In the United States, it's roughly a disease that has a prevalence of fewer than 60 individuals per 100,000. I think it's around 58 right now, actually. And by definition, this often requires fewer than 200,000 patients existing in the US that have been identified to qualify as a rare disease. But again, it's a spectrum. There are rare diseases and there are less rare diseases. I know that's a confusing way to phrase it, but this infographic that I pulled, I think it's from 2019, sort of illustrates different rare diseases of differing rarity. So multiple sclerosis is not a rare disease. It's actually above our threshold for definition of a rare disease in the United States, but it has not a lot of patients, but it's kind of a lot for a rare disorder. Narcolepsy, Fabry disease, cystic fibrosis, these are all classically defined rare disorders, and you can see them here. But at the extreme left side of the screen, for those of you viewing it, is Cushing syndrome. That sort of gets into the realm of these ultra rare disorders where there's so few patients, oftentimes below 3,000 to 5,000 patients, and definitions are completely arbitrary. I like to use any disorders around that 3,000 to 2,000 threshold as defining the ultra rare space. And these disorders have even more challenges facing them because the few patients that they have in their disease often is not compatible with the current commercial design for pursuing disorders that we have in the biotech space and the biopharmaceutical space right now. So that's an exceptional challenge facing them. And oftentimes, if you are a commercial biotech company and you're working on a rare disease, you are incentivized by the policies put in place to work with diseases that are closest to that 200,000 patient population. So that's where the economics and policy sort of come into play. You get the same rewards essentially for working in a disease population that has 199,999 patients as you would a disease that has 10 patients. But that is why we don't usually see development efforts at the extreme left end of the spectrum. So I just wanted to clarify that. But I do want to also inject some optimism into the conversation too. I actually don't think the major bottleneck to developing therapies for these rare disorders any longer is scientific. You heard the story of baby KJ earlier today, if you were online for the morning session. I think that has effectively put to bed that we can actually produce these genomic medicines on demand. And there have been extraordinary advances over the past decades in both genetics, genomics, and diagnostics. So we can sequence people, we can identify diseases, we can diagnose specifically what's wrong. That's step one, and you have to find the target that you want to treat. And now we have an expanding stable of genetic medicines, from gene therapies to cell therapies we can that are not just cool science experiments. Some of them are validated and clinically approved products. So we have this expanding array of genetic medicines, an enhanced ability to diagnose them. So the bottlenecks we had faced in decades past have largely been resolved on the technical and scientific side, but we still have significant barriers remaining, and these are often structural and not scientific. So I'm listing a few of the barriers here that I see constantly in the rare disease space. Number one is economics, right? There's a high cost for developing a medicine. And this often comes because of the demands for producing and manufacturing the medicine and characterizing it, chemistry and manufacturing controls, the CMC conditions are very expensive for some of these more modern modalities like cell and gene therapies. And if you're developing these for very, very small populations, the cost can be pretty staggering. And so it's really hard to develop those medicines on a cost model, on a profitable cost model for very, very rare diseases. So that's a huge problem in this space, probably one of the biggest. But again, that's a structural issue. On the science and translational side, there are risks, but these can be de-risked. And I will say these vary according to the disease. And this is usually limitations by our knowledge of biology for that particular gene or pathway, and then the availability of the tools to interrogate drugs for that disorder. These could be mouse models or cell-based assays or biomarkers, other things we need to actually put together a proper drug development program and proceed to clinical trials. So this can be a risk, but it's a surmountable risk in my opinion. And rare disease by definition is rare and understudied. So these models may not exist at all. And this can be a bigger barrier for foundations and parents and families that are newly diagnosed and don't have this framework of science and tools and assays to build upon. If it's been built for them yet, they have to establish it. So that's one barrier that I see, but it can be surmounted. The third one is regulatory and evidentiary. Our current framework for conducting clinical trials is largely to support big diseases and diseases that are being treated by sort of older modalities, small molecules, antibodies, things like that are very well precedented, and we understand how to make them, we understand the variability, we understand the risks of them. Regulators definitely need to embrace more flexible evidence packages, and they have. The FDA just recently released 3 draft guidance updates related to cell and gene therapies that allow for additional considerations in clinical trial designs and endpoints. So this is great. There are things happening in this space, but more needs to occur, and I'll get into that a little later. On the manufacturing and supply side, again, I mentioned the manufacturing costs are enormous for something like an AAV gene therapy. And if you're going to develop this to be dosed into, say, 10 patients total, that's still a $3 to $5 million cost to manufacture this, characterize it, and then distribute it to the patients that need it. That's an enormous financial barrier that most companies will not want to take on, or even academic institutions. And so that burden largely falls to patient advocacy groups to support this piece, or to find partners to help them do this in a sort of consortium manner. But these are the 4 major barriers as I see it in this space. From my position working with a lot of rare disease foundations, I'm seeing more and more patient advocacy groups actually driving the preclinical research and usually even carrying that forward into the IND enabling studies, or even in some instances, the patient advocacy group becomes the sponsor of the clinical trial itself. There are rare occurrences of this happening, but I foresee this becoming more of a norm. And this is largely due to a retraction of the traditional commercial companies in the cell and gene therapy space from the for-profit sector. You don't have to go very far to read the trade articles and stat or endpoints to see the big name companies withdrawing from gene therapy or cell and gene therapy research. So this has led the patient advocacy groups to sort of pick up the slack because they don't have a choice, right? They want to develop medicines for their loved ones. So the burden of developing these programs and carrying them forward, sometimes into the clinic, can often fall onto the patient advocacy foundations themselves. But they have partners in this space. There are academic labs that have exceptionally smart researchers that know the biology of the disease or the gene itself or the pathway and have tools available. Oftentimes these labs can do some of the enabling work, but they do need guidance. Not all of these academic researchers have worked for a pharmaceutical company before or done a drug development campaign themselves. So additional support is needed to ensure that it's done in manners consistent for IND's submission and application. There's also a significant bridge on funding. Nonprofit philanthropic funders, oftentimes venture philanthropy, can fill the void, but this gets to be a challenge for very small diseases too, because your donor base is small, because it's just very small disorders. So that can be a challenge to fund this in a venture philanthropy model, which might have some sort of commercial return. Oftentimes I see more mission capital or people that are just care about the disease and don't really require the returns of a venture philanthropy route or the traditional ROI associated with commercial entities. And then oftentimes what I see is academic research will progress to a period within a research institution, but it sort of stalls out at those IND enabling studies. This can involve GMP manufacturing, which again can range from $1 to $5 million, or running the safety toxicology studies in large animal models and rodent models, which can also run into the millions of dollars. So these are high dollar cost items that often that's where the program stops, unless someone can fundraise to pick up the slack on that end to bring it forward. But I do see patient advocacy groups stepping into this space and figuring out creative ways to fundraise or access grants to get over this. There are incentives in place that should help enable drug development in the rare and ultra rare space. The first one is the Orphan Drug Act and Orphan Drug Designation. This is a U.S. policy that applies to all rare diseases, and that's a rare disease again is any disease with fewer than 200,000 patients in the United States. So that can be very less rare diseases and then very, very rare diseases, the ultra rare side. It helps to decrease some of the challenges in the regulatory space, and it also gives some additional incentives to developers, longer period of exclusivity, waiver of user fees, which can run into the multi-millions of dollars, and also accessibility of grants. So this is one incentive that is here, but usually sponsors tend to fall on the less rare side of the spectrum, closer to 200,000 patients for the disease indications that they tend to pursue here. But it has a benefit to all those rare conditions. The second one, and this is kind of a mixed bag approach, we had some sunsetting for one of the biggest carrots in the arsenal here for rare disorders, is the rare pediatric disease designation and the priority review voucher associated with this program. So essentially, if you're developing a medicine for a rare disease that also impacts children or those under 18 years in the United States, you can get additional incentives. And the biggest incentive that the RPD-PRV had was if you took a drug all the way to full approval, you would be granted a priority review voucher. Now, this could be used for a faster review of a second drug. And these PRVs actually have a tangible value associated with them. You can sell them. You can look this up on Google. There have been many PRVs taken, granted to sponsors who developed a medicine for a rare disease, and then they sell it on the open market, usually to one of the larger biopharmas, for values oftentimes exceeding $100 million. Now, the fortunate aspect of this is that the program has officially been sunsetted since December 2024, but there are ongoing conversations, and hopefully through what we hope is a soon to be passed continuing resolution, perhaps this could come back. But under the standards set today, sponsors that have rare pediatric disease PRV designations must get their drugs approved before September 2026 to get that PRV award, unless it's renewed. I hope it's renewed. Cross your fingers for that. Another sort of incentive in this space sort of comes from a newly created entity within the FDA that doesn't play a role in the regulatory process, but does play a major role in coordination for rare disease disorders between CDER and CBER, the two big groups under the FDA that approve medicines. One is biologics, the other is drugs. They're a centralized point of contact for sponsors to interact with, and they're increasingly playing a role in putting forward more flexible regulatory endpoints or biomarkers and outcomes associated with clinical trials. They do run a workshop series called the RISE Workshop, and it's oftentimes socialized by the Reagan-Udall Foundation. So I'd urge you to go explore these. And in a recent RISE workshop, they put forward this new program called the Rare Disease Evidence Principles Program. This adds an additional incentive to rare disorders that have fewer than 1,000 patients in the US and are very severe. That means they're facing progressive functional deterioration or a significant risk of disability or death. This program offers a very flexible single-arm clinical trial, no control arm, and has some flexibility on the endpoints for approval that can be based on real-world experiences or patient-reported outcomes. So it's an indication that the FDA has appreciated the challenges facing that rarest end of the rare disease spectrum, and they're being responsive to it. So it's a step in the right direction, but I don't know that it's the overall solution that's needed in this space. So you can stack these incentives, right? You can have the orphan drug designation, rare pediatric disease designation, and RDEP if your disease is fewer than 1,000 patients in the US. And if the PRV is restored, this might have an additional monetary incentive associated with it. This could incentivize developers to want to get involved in disorders that are at that fewer than 1,000 patients, the rarest end of the spectrum, which is good. But putting a sort of patient number threshold on this is a challenge, because what if your disease has just above 1,000 patients? That's unfortunate. A tiered incentive approach would be a more practical solution to this, to help right size the incentives associated with the rarity of the disease population. So I hope that something like that will be put forward in the future, because if I put my commercial drug developer hat on and I see these incentives, I might be incentivized to go after disorders that have 999 patients or 800 to 1,000 patients. And maybe I won't want to go into the patient populations that are extraordinarily low again. So again, we might have the same issue we've had with rare pediatric disease designation and orphan drug designation, where it's like the commercial developers want to target the largest end of that spectrum to capture those incentives. But we'll see how this plays out. It's a very new incentive that was just released in September of 2025. So we'll see how it plays out. I'd also like to see the Rare Disease Innovation Hub play a bigger role in perhaps creating a regulatory track for these commercially non-viable diseases, the ones that are so small, that are often led solely by a patient advocacy group or an academic sponsor or a partner in those efforts, or some biotech companies even. That would actually help, where the FDA could play a role as a partner in developing these medicines. And I think there would have to be some other strings attached to this, that drugs would be only for the public benefit. There wouldn't be above a certain threshold of return on investment associated with the willingness of the FDA to play more of a partner role in this. But I don't know what that would look like, but that is something that's definitely needed in this space for these rarest of rare diseases, because right now it is just all put on the patient advocacy groups to do most of the work. And if they can find a partner, that's great, but we need to incentivize more partners to help them in this effort, so they're not carrying that burden by themselves. I do want to call out a few interesting models, and these are not selected to say these are the only companies, but there's just a few different companies that have done some unique things in the rare disease space. One is Therapeutics. This was a company started by a rare disease dad out of Canada, Terry Pirovlakis. You might have read about him if you've attended Global Genes or NORD or Every Life Again, any of these conferences. He developed a gene therapy for his son, Michael. Melpida is what they called it. And he's actually spun this out into a company. It's an AAV9 gene therapy company that focuses on CNS disorders, but it's been structured as a social purpose company. And his only goal is to generate enough money to treat more diseases. And that's his approach. And he's done this through some creative collaborations with the Bespoke Gene Therapy Consortium. This is a public-private partnership, a government public-private partnership, where they're working on making gene therapy more accessible to more disease groups. They've also explored some funding mechanisms local to California, the CIRM, the California Institute for Regenerative Medicine, to sponsor some of their drug development efforts and clinical trials. The second one here is Mozi Therapeutics. They're actually a rare disease company that focuses on extraordinarily rare diseases, and they have some venture backing, which is unique. They advocate for a very strong patient-scientist partnership model, where they can leverage a lot of the learnings that are based out of these really savvy patient advocacy groups. And I think it's a model that many other pharmaceutical companies need to model. and actually build up their patient advocacy groups to not just focus on reaching out to patient groups at the clinical trial stage for enrollment, but actually even at the earliest time points of drug development efforts, involve them in the preclinical space. The third company here is Gemma Bio. This was a company started by Jim Wilson, formerly of UPenn. He's actually taken the approach of funding his company through agreements with other government entities. The country of Brazil and Abu Dhabi, he's actually structured agreements with them to do manufacturing for some of these complex biologics. And that's what's helping to fund his company. So there are some interesting company models and funding sources that are being pioneered. I also wanted to call attention to two unique funding models in this space. One is the Buffalo Initiative. This is it's sort of a CNS-focused drug development fund, largely driven by patient advocacy groups, parents of rare disease, and they're targeting philanthropic and impact investors to invest in this fund and develop medicines in these rare diseases with a very modest return on investment, much lower than the commercial venture-backed models are supporting right now. So this is one interesting model that I hope is successful. Another one is the Orphan Therapeutics Accelerator. This is seeking to take deprioritized clinical assets from larger companies where they've decided to pause a trial because maybe it's not going to meet the profit threshold that's needed for that company. But they're trying to take these assets forward, these deprioritized assets forward, under a hybrid nonprofit and for-profit model. So that could be another model which enables development in this space. And then I do want to call out the baby KJ story that was spoken about earlier in the morning show, because I think this shows that gene editing in the liver might be amenable to sort of a platform approach where maybe you're not getting an IND, doing an IND submission or asking for IND approval on an indication by indication basis, but maybe it's more of a platform approach where it's sort of on demand. And this could be modeled off of another biotech company that's in the space, BioNTech, that's currently sort of has this on-demand mRNA cancer vaccine approach where they'll actually take a patient biopsy, send it to the lab, do some sequencing on it to identify neoantigens, which might be patient-specific, and then construct a specific mRNA sequence that has all the neoantigens in that patient's tumor, create that genetic medicine, send it to the patient, and they get dosed with it. This could be a platform approach that is applied to many of the gene editing modalities. And so it might be, in the future, maybe a platform approach could be accessible for some of these gene editing approaches. I mean, delivery to each of the tissues is something that still has to be resolved. I think we can deliver to the liver fairly well, but to the bone marrow, it gets trickier, to the brain, it gets trickier, but this is one approach. And then there are some other ideas being put around by innovators in this space, Dr. Kieran Musunuro from UPenn and Winston Yan from the Ana1 Collaborative. They've spoken publicly about thinking about some of these genetic medicines less as a drug and more as like a complex surgical procedure or an organ transplant. You don't go to an IND submission process or application for these complex surgeries. It's overseen at the hospital level by regulatory bodies or other non-government institutes that sort of accredit and ensure that these processes are done in a professionalized manner and done correctly. So this could happen probably in the very far future once we understand gene editing far more in detail than we do now. But this could enable us to reframe the usage of these genetic medicines and think of them less as like a drug and more as like a molecular surgery where we're just allowing the clinical team to select the right tool to fix that patient's mutation. So that's another type of model that could manifest at some point in the future, but I think that's going to require some additional unlocks to occur. All right, I talked about, oops, I went back. I did want to just say there's a couple things that foundations and academic consortia could do to help enable this future to come about faster. I think academic institutions need to start sharing and centralizing CMC and GMP production facilities and enabling their access and sharing of these resources to patient advocacy groups, because I don't think each patient advocacy group should have to go to a CDMO, negotiate those costs. I think we need some hubs to be built up where patient advocacy groups could go to this one place. They could manufacture for a lot of different diseases, a lot of different indications using like a certain modality, I think that would help unlock some of this challenge around the manufacturing side and enable a single site where patient advocacy groups could go to make these medicines. The usage of natural history studies, registries, and real world evidence to have smaller trials or to have lower powered trials where we can more adequately understand how well these medicines are working is something that we need. And then the regulatory engagement and innovation endpoints. We need sponsors and regulators to be less adversarial and more of a partner, especially when some of these sponsors are patient advocacy groups. They really need the support of the regulators because they don't have the massive resources that a biopharma organization would have. And they are the ones carrying these things, these therapies forward. So they need more support. And I think if we employ some of these coordinated mechanisms, I think we're more likely to see successful outcomes. And I think on the funding side, we need to explore the usage of sort of mission capital or aligned investors that have a unique connection to those disorders. Pooling patient registries, sharing assays, sharing resources is something that I think would help to bring down costs across rare disease in general. And we also need to not have all the universities making their own CAR-T facilities or all the universities making their own gene editing facilities. I think centralizing them and sort of choosing a specialty amongst these different universities would help sort of defray those costs rather than everybody trying to build everything themselves, forming more of a consortium there. And then global regulatory communication is going to be needed because rare diseases don't usually respect international or geographic borders. They happen globally. And so we're going to need to be able to do this throughout the world. So just some quick takeaways. I know I'm a little bit over time, but some quick takeaways. Foundations really should invest in high quality natural history studies and registries. They need to understand their disease and what outcomes they'll assess. Funding models, they need to go after some creative funding models where they can work with partners, access in-kind services or reduce cost services, perhaps a cost plus model where they're just paying the cost of reagents plus a teeny bit extra, but not necessarily what the big pharma companies would pay. Negotiating access forward agreements to ensure they have access to anything they help develop and help get in-kind contributions for the manufacturing of those therapies with their partners. The usage of multi-disease consortia to leverage platforms or assays that might be common to specific points in which these genes play in the pathway or biomarker outcomes, that would help to reduce every foundation needing to build these things themselves. So I think trying to cluster together similar diseases that might have similar endpoints is definitely something that should be done more frequently amongst the patient advocacy group foundations. And then some endpoints for academic researchers. I think thinking about this as a drug and less as like an interesting biological question to solve, that really requires a mind shift away from the traditional academic mindset of publications and grants and novel investigations, and really doing the more boring drug development work or thinking like along those lines. But that is what's needed in this space. Increased use of consortia, standardization of SOPs would definitely help in making this a little more achievable and accessible from the foundation side and all their partners they interface with. Aligning with your patient advocacy group's priorities in terms of what outcomes they would want to see in their patients. It might not always be what you would think based on the reported literature. So talk to those parents and those patients with the disorder. Grant mechanisms that fund IND enabling work, and by this I'm referring to the manufacturing, the GMP manufacturing, and the safety toxicology studies, those high dollar cost items. We need more grants specifically targeted at that pre-IND bottleneck space. And then, oh, these are speakers notes. Those weren't meant to be in there. I apologize for that. So we need a better partnership between academic researchers and patient advocacy groups. And I should have also put on here engagement with the tech transfer office at the university. They need to understand the difference between a huge chronic blockbuster disease that's a massive disease population and a rare disease. Right now, the lawyers within most tech transfer institutions structure agreements identically across those 2 points. And I think that creates a lot of challenges and complications for the patient advocacy group partners. There's some more interesting stuff in a publication that I co-authored with my friend Juan Carlos Lopez, and more information you can find out at our foundation website here. And I'll pause it there and turn it back over to Eszter. Thank you.
Thank you. Thank you so much. That was a lot of inspiration, new things for us to think about. I think a lot of these concepts and questions are fairly new to our community, but it's essential to think about them and plan for success. So if the community has any questions, please type them into the chat window. What stood out to me, Joe, in your talk is that it's really about coordination of the work. It's not simply hoping that the right people will collaborate at the right time, but somebody needs to look at the whole picture and figure out what's going to be missing next, or what is it that will accelerate the right processes at the right time and do them. So I think it's a lot about coordination to make this entire thing work, because what we really want is therapies in patients. It's really not sufficient to stop anywhere before then. So let's see. I don't see any questions in the chat yet. So maybe I should think of some.
Yeah, I'll just echo your comment there, Eszter. Like, I do think most patient advocacy groups have to construct all the partner lines that a biotech drug developer has, but in a decentralized manner, and find the right partner to help them do that function, whether it's manufacturing, whether it's running those safety toxicology studies, or helping them do the regulatory filings. It's hard, and they require multiple partners with multiple different skill sets of expertise. So I think it's a lot of coordination. It's a lot of identifying who's the right partner to help you do the thing that you need done next. And it's just really, it's a tough process to navigate. But I do see more and more foundations successfully doing it. So I think those learnings are being leveraged by the rare disease community. And I think there's going to be more examples of this occurring. I think it's going to become more common.
Yeah, and the other key word here, I think, is the partnership, because we can, as a rare disease, we can no longer rely on somebody else doing it for us. We need to be active. We need to have an active seat at the table and drive and move things forward as needed, because sadly nobody else has that drive and need to really help our children as much as we parents do.
But that makes you the ideal partner for any biotech company or academic partner that would be interested. I don't know another more motivated partner than a rare disease parent. So I think it's the partner of choice. So it is kind of a superpower that most patient advocacy groups can lean into.
And that is our only motivation, is really to help our kids. It's not about making money or be profitable for ourselves. We are interested in responsible financial management in order to be sustainable and be able to go all the way to therapies and treatments for our kids. So we are the ideal partner, and I hope more and more other partners in other fields will understand that and join us too.
Yeah, that's my hope as well. I hope biotech and biopharma will take greater note of these really savvy patient advocacy groups that are really sort of firing on all cylinders, advocating for their patients, doing registries, also fostering drug development efforts. But I do see this trend increasing, so I suspect it will be hard for the biotech industry to ignore this trend, and they do it at their own peril. So.
Yes, indeed. And yeah, I agree, actually. The biotech companies that are familiar with the rare disease field, they tend to be more open to these sorts of discussions and partnerships. I think academia is lagging behind in some areas, there, but I've seen changes there too, where we are now able to have these kinds of conversations of how we can truly partner and move things forward together.
Yeah.
All right, so I don't see any specific questions for you, Joe, so thank you so much for joining us on this Sunday.
Thank you.
A rainy one at that. And we can move right on to the next topic. So that's me. And just to recap from earlier today, the problem we are trying to address here is SDS. SDS is a serious life-threatening condition. And as we all know, it causes a high risk of developing leukemia, which is very, very hard to treat. And it's estimated to hit about 30% of patients by age 30 and more after. I think the most recent estimates are 50% by age 50. So this is the problem that we are looking to solve. And so our mission is to save lives by accelerating therapy development, or in other words, to give SDS families more birthdays to celebrate. That's our sole focus. A more immediate or specific goal for us is to get to clinical trials for SDS by 2030. And our vision is that this will actually help real patients, right? But also just by demonstrating feasibility, it can then also open the doors to other therapies as well. This will lower the entry barrier for other therapies as well. It's very unlikely that the first therapy will serve all the patients and cure them all, right? The first therapy will probably be helpful to a subset of patients at a certain stage of disease. And so we do need to keep an eye on the bigger picture and ensure that we enable additional projects to develop over time at the same time. So going back to the ideas we discussed with Joe just a few minutes ago, we talked to dozens of successful rare disease organizations and analyzed what they do. We also got support from the Chan Zuckerberg Initiative's Rare as One program and countless experts in the field, such as Joe. And in essence, what we need to do is coordinate all these efforts to take us from basic research, as they call it, that happens in the lab, all the way to actually helping our children, and that our children also includes adult patients. And so what do we need to coordinate? We need to identify the tools that are necessary for this translation. We need to fund the infrastructure, and we also need to coordinate funding. We also have a lot of help from our SDS expert advisory board and our strategy and method expert advisory board to develop these ideas and strategies of what we're going to invest in. And investment in this case doesn't only mean investing money, but really our time, effort to build those connections and partnerships. So when I think about what we are doing and what we want to keep on doing in the future, we can divide these activities into kind of three buckets. The first one is building and sharing research tools. So again, it's not enough to just build a research tool that is then only going to be restricted in one particular lab, but we want to build them and make them available to any researcher who might need it. So research tools include things like mouse models, iPSCs, organoids, biobanks, but also data, access to data, and developing biomarkers. Then the second category of things we need to invest in are infrastructure that will enable clinical trials. So things like doing the PFDD meeting that hopefully many of you have attended earlier this summer, which builds conversations with the FDA for them to understand what it's like to live with SDS and what type of therapies and changes would be meaningful to our patients' quality of life. Clinical trial matching. So for us to be able to inform the community when new clinical trials become available so that recruitment can happen reliably and effectively. Precision diagnostics, like we cannot treat patients unless they are accurately and precisely diagnosed, unless they have the accurate genetic testing and they know exactly what they have. And then we also need to have an understanding of what a patient journey looks like in real life, just in the healthcare system. And to be able to learn about that, we need ICD-10 codes, and we'll talk about that in a minute. And then the third bucket of activities is the actual funding, and we call that investment. So we want to strategically invest in the right projects at the right time for the right reason and purpose, which is oftentimes catalytic, meaning that we don't expect to be able to provide $1 billion in funding to take something from the start to the complete finish. But we envision that by funding maybe several $100,000, that might open the doors to larger funding opportunities. And hand in hand with that is also the mechanism of how we want to fund some certain work. We want to be very clear about what the money should be used for, what milestones we want to see, what the time frame should be, who is going to own the IP, the patents, if there is a partnering happening in the future where the institution receives a huge amount of funding from a commercial partner, should we be able to recoup a little bit of our investment so that we can reinvest that into other projects going forward to keep the momentum going. So those are some of the ideas that go into the strategic investment piece of it. And then another aspect of this all is that when we think about impact, like why are we doing it? The impact we're thinking about is not how much money we have spent, but really the benefit that we see. Sometimes we don't need to spend money at all, and we see a huge impact of our efforts. For example, the building a public biobank. So we have a biobanking effort in partnership with the Coriell Institute. The NIGMS repository doesn't charge patient organizations anything. And to create our iPSC cell lines, we received a matching grant from UPenn. So again, our monetary investment was modest. But now we know that several academic labs and several companies have requested SDS patient-derived cells to explore whether their programs or whether their techniques, technologies could be applicable to the disease. So that's the impact of that effort. Then what about the data? So we started a program called SDS GPS, which is a global patient survey and collaboration platform. So this is a program where patients from anywhere in the world can enroll, go through an informed consenting process, and then fill out surveys about their experience with SDS. So we exclusively focus on patient-reported data, and it's available from anywhere in the world. It's translated or available in 5 languages, possibly more to come in the future. The surveys are validated, which means that they're standardized and researchers know how to use them and interpret the data. And then it can also be compared to other diseases, for example. And so this is data that the patients own. Like if they don't want to participate any longer, they can withdraw their data. If they want to compare their data to other patients, they can see that in the system too. And there's some other nice bells and whistles in there. But so far, we enrolled over 70 patients from over 15 countries. And so that's a great start, but we hope to for more of you to participate. To participate, it's really easy. All you need is about 45 minutes or so of time. If you have your genetics report that led to your SDS diagnosis, that is also super important. If you have it, please upload that. And then you just need internet, and you can use your phone or a computer, laptop, any device to enroll. And all the information is on our website at SDS Alliance /SDSGPS. I'll share that information out again. Now the impact of this is manifold, but most of two benefits just off the hat is that if we learn about clinical trials that are coming out, we can inform the patients that they are out there and they should explore, right? Because we will know if they have a certain set of symptoms or a certain genetics, we will know what trial may be relevant or of interest to them. Another benefit is that we can talk to researchers in all areas, could be pharmaceutical company or academic, and really represent what our community as a whole considers meaningful and important when it comes to SDS. We can discover patterns that maybe haven't been known before, like are there issues with the eyes, for example? If a lot of patients start reporting that, that may be something new for us to explore as well. Whereas in other data collection efforts that maybe just focus on the, say, the blood issues, they may not necessarily notice those kinds of patterns. So this image here is actually a poster that we presented at the SDS International Congress last June. And so it was an opportunity for the academic researchers to see what SDS patients report regarding their experience with SDS. This is also linked on our website if somebody wants to zoom in and look at all the details. Yeah, I hope you visit, and I'll share the link later. Now to the second category of activities, which is the infrastructure. I mentioned already the PFDD meeting and the Voice of the Patient report are some of the key impact tools. We had just in the first month, we had over 1,500 views of the PFDD meeting that we posted on our website. And I think most rewardingly, we had a meeting with the FDA after the PFDD meeting, and the staff that attended our meeting reflected back to us what they learned and heard. And they really, really heard us. They heard that there are all sorts of day-to-day impacts with mobility and pain and digestive issues. And at the same time, that we as a community are very worried about the risk of leukemia and what that means for our family. So if in the future a therapy comes across their desk that could reduce the risk of leukemia, they now understand that even just this reduction of leukemia risk without addressing any of the other symptoms, that in itself may be a meaningful change for us. And so that can help move future therapies forward. Another success, impactful success, is the ICD-10 code. So we successfully advocated to create an ICD-10 code for SDS in the US. It's D6102. It went into effect in 2 years ago. And the benefit, I mean, a code is really boring by itself. But what that means is that now if somebody searches the electronic, like the whole big database of all the electronic health record, they can now pick out the experiences of patients with SDS. So this is not possible without an ICD-10 code, and it is now possible. And so in effect, a code made the patient journeys visible for over 650 patients so far. And then now to the third category. So this is the newest category of investment for us. And this is also something that we will need the help of the community to move forward. But just one aspect of it is that when we talk to academic research groups or even industry, we just want to be really clear of what we're funding, what the partnership will look like, so that the expectations on both sides are clear, and there are no roadblocks on moving things forward. Like everybody's clear of what each party's responsibility is, and that can really open doors also for future collaboration as well. So back to our current goal, right? We want to enable clinical trials by 2030. That's our goal. So if we go back to that, and we want to think about how to actually do that, now that we built all the infrastructure and the research tools, or at least started to enable them, we now also want to think about what are we going to fund next and how. And to make those decisions, we analyzed the landscape of what type of therapies are actually out there or in development. So I don't know how small this appears on your screen, but this is the timeline of drug development, again, starting with drug discovery and lab, preclinical work, through the IND, and then clinical trials on people, phase 1, 2, and 3. The top half of the screen focuses on really SDS-specific therapies, and the bottom half on symptom management type of therapies. So let's start from the bottom, actually. So there are efforts around reducing the toxicity for conditioning when it comes time for transplant. So many of us know about treosulfan. It's a conditioning drug that seems to be working better for SDS patients than some other drugs, and it has been recently approved for non-leukemia applications. So it was approved, I think, about a year ago or so, or earlier this year. And there are further trials now to see whether the drug is also helpful and effective for patients when AML and MDS are already there. And then there are academic efforts around milder conditioning regimens, which use antibodies, for example. So I'm not aware of anyone having tried that for SDS specifically, but there was a trial for Fanconi anemia, which is very interesting, out of Stanford in collaboration with Jasper Therapeutics. So it's just something to keep an eye on to see if it will progress. My understanding is that the results are good, but the economics might make it challenging to move the project forward. And then there are some I touched upon X4 Pharma's drug for neutropenia. That's actually currently in the phase 3 clinical trial, and they're looking for SDS patients who are, I think, 12 and older with certain criteria. I can send any information about it if you like, or you can look it up on ClinicalTrials.gov. And then there is a new company that just emerged from stealth mode called Eternal Bio. They're looking to develop a cancer vaccine that would target p53 positive clones in SDS before they transition into leukemia. So now to the top half of the screen, so that's the SDS-specific drugs. And I divided them into 3 categories. There's the DNA targeting, so gene editing, gene therapy, prime editing. There is the RNA targeting work, such as Ataluren, as we discussed with Dr. Bezzerri and Cipolli this morning. There were some efforts around ASOs as well, but I don't have the latest information on that. And then you guys are probably also familiar with Dr. Warren's work in Cambridge on small molecules. And there's another company that is working on small molecules. So they are all in this preclinical stage of work. And it's not clear how fast or whether they will be able to progress to IND. So for today, this is what I really want to focus on, and specifically the top of this graph. So we think that the prime editing project by Dr. Bauer at Boston Children's Hospital is the furthest along, is the closest to IND enabling study. So we believe that this work is at a stage where our modest investment can help them move across the this finish line to get through to IND, not by us single-handedly raising $2 million, but by finding the right partners, additional grant opportunities, philanthropic partners, and everything in between, like supporting them in applying for bigger grants successfully. So all that, we want to focus on helping them really move into IND to phase 1, because A, it might help patients right then and there, and it would open the doors for other therapies to advance as well. The project by Dr. Elefanti in Australia that we covered earlier today is also very interesting, but it's at an earlier stage of development. It's a little further away from IND and phase 1 because it's a newer technology that hasn't been approved for other therapies yet. And then we have extensively discussed the status of Ataluren for SDS as well. All right. So to summarize, there is really strength in numbers. We want to get to clinical trials by 2030, and we as patients can contribute in 2 very important ways. One is just joining the community, learn about research, and contribute your data wherever you can, like joining the SDS GPS program and all other registries, donate samples, and think about participating in clinical trials and what goes into it. And then the other aspect of it is help us fundraise and invest in research with funding. That's another area that the community can contribute to. But it's not gonna be the only way that will take us all the way to all the funding. So I just wanted to emphasize that I don't want anybody to feel pressured that they must fundraise, but it is an opportunity for those of us who are in a position to donate or fundraise that can really make a meaningful impact as well. I'm gonna skip this slide just to say that we are basically doing each and every bullet point of this chart. So I felt very validated and seen when Kendall presented this. And so our goal is to ensure that your support has the biggest impact, that we reach our goals together. Leverage your contribution. In the interest of time, I'm not going to go into great detail of this, but I can share it later. But this is a summary of what your support has helped us achieve over the 5 years. So SDS Alliance was established in 2020, so just 5 years ago. We started out with a mouse model project that is still ongoing because biology. There were some unexpected things happening with the biology. The project slowed down in the middle a little bit, but now it picked up again, and we're still pursuing generating a transgenic mouse model to enable all sorts of research and drug development efforts. Then we have the cell biobank in collaboration with Coriell. That's alive and well, but we are looking to expand it with more iPSCs and easier access to cells. We got the ICD-10 code. We started the collaboration with Australia, which will give us another type of mouse model too. We have patient-reported data collections going on through the SDS GPS program. We have the PFDD meeting and a lot of other engagements with the FDA. So actually, when Joe pointed out that there is this new hub for rare diseases, we participated in the launch of that and had the opportunity to speak to what we would like to see in such a center. We have several blog posts on our website about those types of interactions. And then so the most recent effort is really figuring out how to invest wisely into the translational research projects. And to enable all this, again, this is all because of you, the patient community, that made all this possible, because we were able to go to bigger funders and say that our patients are here for it, they need this, and we have a whole community behind us that wants to see therapies come to fruition. So we got in 2021, we got the CZI Rare as One grant. Then we got leadership support from the Milken Institute and the partnership with UPenn to build our iPSC biobank. And more recently this year, we got a PCORI Engagement Award to help bring the patient voice into all stages of research and drug development, and specifically to comparative clinical effectiveness research, CER. So thank you all for enabling that. Now, if you are interested in donating and fundraising, go to our website, sdsalliance.org. There's a donate button right on top. If you have the means to become a monthly donor, that would be amazing. That really shows that you support our work on an ongoing basis. And if you work for a company that offers donation matches, that is, of course, also helpful. They often double your donation, and there is a link to how to check that out on our donation page as well. And if you're interested in fundraising, again, on the donation page, there are links to tips and tricks on how to fundraise, and also how to set up a personalized fundraising page that highlights your family. And I'm happy to help customize those websites or help with any of the efforts really to create marketing and communications materials to help you be successful at fundraising. There's one fundraiser I want to highlight So there's two kinds of fundraisers. There's one that we organize, the SDS Alliance organizes, and then there are those that families organize independently to benefit the SDS Alliance. So the only event that we ourselves organize, the SDS Alliance, is called the Million Steps Closer to Cure SDS. This year it's gonna be in the first week of November. This is an annual event. 100% of the proceeds go towards research and towards our goal of clinical trials by 2030. There are T-shirts, all sorts of things. So I recommend that you check out the website. It describes all the details, sdsalliance.org/steps. And then we can also help you create a fundraising video. So we can help you customize flyers, fundraising pages, everything. And then more recently, you might have seen our fundraising video. We are able to put in your child's or your family's photos into that video and maybe shorten it a little bit. But it explains why we need help and how we use funding. So I think actually, Jeff, now might be a good time to play that video.
Investing in a rare disease can be very rewarding because you will know that the gift you're giving to the organization will go as far as it can and will impact lives today, tomorrow, and for years to come. My name is Eszter Hars. I am a scientist and researcher. I have a Ph.D. in molecular biology, and I am the founder, CEO, and president of the Shwachman-Diamond Syndrome Alliance, or SDS Alliance for short. Most importantly, I'm the mother of a child with Shwachman-Diamond Syndrome. As parents of a newly diagnosed child, I was wondering, why is there no therapy yet? Why do we just have to wait and see and not be able to do anything in the meantime? That quickly changed for me into, well, what can we do? What can we do as scientists? What can we do as parents? What can we do as a community? This is what catalyzed the formation of the Shwachman-Diamond Syndrome Alliance. Our goal really is to accelerate research and developing therapies fast. When my daughter was diagnosed, I joined the Facebook support group, and nobody knew how many adult patients are there. Will my child graduate from school, go to college, get married? Once I started the SDS Alliance, I partnered with an adult patient, and together we identified over 100 adult patients, and that by itself was huge for me. Shwachman-Diamond Syndrome is a rare genetic disorder that causes a lot of problems in the body. Most importantly, it causes problems with the bone marrow.
SDS is unique in many ways. One of the reasons that it's unique is this very high risk of developing leukemia. Because of this risk, patients that we see with Shwachman-Diamond syndrome need to undergo routine screening for the development of leukemia, and that's a hard thing to live with.
Current treatment options for leukemia are unfortunately not very effective. Typically, it's bone marrow transplant or stem cell transplant. The problem with that treatment in SDS patients is that patients cannot tolerate the standard treatment that would be necessary to treat the leukemia. So patients either succumb to complications from the treatment itself, or leukemia relapses, and that's what leads to death. Current treatment options for leukemia are not working. Developing a therapy is much like building a house. It's not just about collaboration, it's more about coordination of the work. If you want to build a house, you absolutely need a developer who would coordinate the funding and all the contractors. They might find the best roofer, the best framer, the best crew to build the foundation and coordinate all the work so that ultimately they end up with a house. You also need infrastructure. A house wouldn't be built in the middle of a desert. You need roads, water supply, electricity, and a community to actually want to move there. Therapy development is much the same. For common diseases, you have pharmaceutical companies who coordinate the funding and the researchers and the regulatory processes to get therapy to the market. But in rare disease, this coordination is missing. The market is too small to make it profitable for pharmaceutical companies to invest. And therefore, not much coordination is happening, if at all. And this is where the SDS Alliance fits in. We see our role not only recruiting the best researchers, but to coordinate the work.
Diseases like Shwachman-Diamond Syndrome fall a little bit through the cracks of traditional research funding organizations. And so identifying unique mechanisms and supporting those unique mechanisms to fund research into diseases like Shwachman-Diamond Syndrome is a key function of organizations like SDS Alliance.
When you invest in this work in the SDS Alliance, you play an integral role in changing the future of thousands of children and families worldwide. You are investing in a future where people affected by SDS don't have to live with debilitating fear of losing their lives or losing their children, who instead can dream of growing up, graduating, getting married, and have families of their own one day, where we can simply look forward to more birthdays to celebrate. Join us.
Yeah, so this is the video which we could customize for you. So where we see Declan in the beginning and Declan at the end, and other kids, we could put in videos or photos of your child if you wanted to create a custom video for your community. Before we jump onto Rebecca's presentation, just wanted to answer a question that's in the discussions. So somebody is asking how they could support these drug development efforts to really reach clinical trials by 2030. And to that, what I wanted to say that all our efforts at the SDS Alliance are geared towards that, to make the clinical trials possible. It includes all sorts of infrastructure, and it also includes direct funding of those groups. Now, the reason we are not specifically calling out a specific project for fundraising at this time is that we are negotiating the agreements, really figuring out how much they need, for what do they need it, what are going to be the milestones, the deliverables, and then we make the financial commitment and pay at the negotiated schedule. So to make a long answer short, we are financially supporting and also supporting these groups and research projects in many other ways. And so if people are on board with our goal of clinical trials with 2030, please reach out and work with us to move things forward. We are basically representing you, the patient community. Our only interest is to make sure that your investment of time, effort, and money goes as far as it can. And that's what we work towards every day and night, oftentimes. So I hope that answered the question, but if not, please reach out and we can discuss it in even more detail. But with that, I wanted to hand it over to Rebecca Hilburn, who is the mother of Declan, adorable little boy we saw in the video earlier. And she has had some great success with fundraising with us and for us, and is willing to share her experience with that. So thank you for being here, Rebecca.
Thank you, Eszter. Can you hear me okay? Yeah, okay, great. Yeah, so thank you so much. I'm looking forward to kind of sharing some of the sort of best practices and tips I've developed over the years around fundraising. But you can go ahead and move on to the next slide. So I'll first start maybe just with a quick sort of recap of sort of how we got here. So this is a picture of my family, and Declan, who is now 8, and Emma is 9. Declan was diagnosed with Shwachman-Diamond at around the age of 3.5. And since then, he has neutropenia. He has neurological challenges. He actually has probably the most impactful issue for him right now is the impact to his bones. So in the past, probably maybe just about 2 years ago now, he had to have a bilateral hip surgery due to what the disease was doing to his bones. And so he's since overcome that and is doing really well, but that's been probably the most impactful. He has some issues where he had to have an eye surgery due to strabismus. But overall is doing really well and very positive and energetic, and a little boy doing all sorts of little boy things. I'll say that the one thing that sort of has really been kind of helpful for me is just really getting as engaged as I can with the community and trying to really get to know families and use and leverage all of the different social media outlets that we have to help speak to other families and share my story. And it's helped me along the way to sort of troubleshoot different things that pop up with Declan each year. So I will say that engaging with the community has been a big help for us. And one of the biggest things really, for me has been fundraising. I've found that it really helps me feel like I'm doing as much as I can for the community and to support this rare disease and to hopefully help try to find treatments and a cure. So really, I've found that over time I've been able to sort of continue to work on telling our story and sharing that out with the larger community and friends and family and colleagues. And I think that over time, as I continue to share our story, I've been able to connect with other individuals who have similar situations, maybe not the specific rare disease that we have, but just other families who are struggling. And it really helps to kind of brainstorm ideas on how to engage more and help and fundraise. So you can move on to the next slide. So here's just some examples in different ways of how you can fundraise. And then eventually I'll go through some of the examples of how we've fundraised over the years. So one really easy way to kind of engage and start kind of small is peer-to-peer campaigns. And this is actually the way I kind of started out, is just creating a flyer and creating a fundraising site. I worked with the SDS Alliance, and just kind of sharing your story and your fundraising details out through social media. That was just a really nice, quick, easy way to kind of do the peer-to-peer campaigns. And I'll talk more about what I did there. But that's a nice, easy, quick way to sort of get your feet wet and test it out. You can also, though, as you start to kind of expand and get bigger, organizing events, you could do something as easy as just getting your family and friends together for a walk. Bake sales are great, anything around food. And then other ways would be connections. So leveraging your community in different ways that you possibly can. I would say we are active with both Girl Scouts and Boy Scouts. So that's a really good clubs that could help to organize events and get the word out and messaging, as well as employers. So making sure that we're leveraging our employers to sort of match the donation. I know a lot do that. And I know Eszter talked a little bit about how there's information out on the SDS Alliance website that explains how to, how to proceed with submitting that request to your organization and making sure that they'll offer that. So what I'm going to do is go into a little bit more details about really how I've sort of executed, I guess, each of these different ways to fundraise. If you want to move on to the next slide. So before so the SDS Alliance offered a few years ago, I think it's been now, sort of a course on how to tell your story. And it was really a great course. I took a lot away from it. And I definitely needed it because I think that's one area where maybe I struggled in the beginning was you really need to get personal with your storytelling to be able to sort of reach and engage and get people to sort of want to support you. So over the years, I've definitely have improved kind of my story about our family's journey and, Declan's journey. And so I think that's really helped me getting personal and, being authentic, obviously, and sharing as much information as you can about the disease and the purpose and sharing updates regularly to be able to show kind of what, that fundraising effort is going towards is really critical. I've also worked with the SDS Alliance over the years in creating posters and flyers, which has been such a huge help because that wasn't really a space that I had a lot of experience in. And so, being able to kind of focus on pulling together all of the other details and executing certain fundraising events and then having their support to be able to do those things that I don't have a lot of experience in has been a big help. And the posters and flyers have always come out to look so amazing. And you'll see some screenshots of these in the following slides. And then the other important detail to, telling your story is making sure that they understand, where is that going? What's the impact? So I think being very specific in when you're, creating, creating any materials, flyers, fundraising sites, right? Just making sure that in that summary that's going out to the community or the peer-to-peer or wherever you're sending it, I think is just really important. So they understand, where is that money going? What is it going to be used for? Is really critical. You can move on to the next slide. So best practices for a successful fundraising event. I will say that I definitely started small, and I think over time I've been able to sort of build on that and organize kind of bigger events. So like I said, I think just kind of getting your feet wet if you haven't done it yet, starting small and just creating a fundraising site, kind of sharing it out to your friends and families through social media. I've been able to raise quite a bit of money just doing that. And it's a very simple thing to do. It doesn't take much time, maybe an hour or so. And then as I got a little bit more confident with fundraising, and I over the years have kind of would sit down and take a day or two to sort of brainstorm and think about, okay, what can I do this year? How can I make this a little bit bigger, reach a little bit more people than I have in the past, has been really helpful. And I've obviously always leveraged my family and friends to help, support that and help me think of ideas. We've talked about being clear about the mission. So how are those funds are going to be used? So that's really critical to include in your, in your story, and make sure that that's really called out in any materials that are going out to fundraise. I've often circled back to any organizations or, local stores, anyone who has supported any of my events. I always make sure that I go out to share just a thank you. Even going as far as to say, we've raised so much money and it's going towards this, thank you again. I think it just really does go a long way, kind of staying engaged with those folks who are contributing to your event. You can move on to the next slide. So this is a great kind of framework that if you sort of follow this, it hits on really all the important points that you want to make sure that you're addressing through your fundraising. Being clear about the purpose, what's the benefits of it? Where is it going? Creating a goal. So I wasn't really doing this in the beginning when I was starting out kind of small. I was just so focused on kind of, doing as good as I can. But then over time, I have found that, kind of coming up with a goal sort of pushes you a little bit harder to make sure that you're doing all that you can to sort of get to that end goal, whatever the case may be. Marketing. There's so many different ways that you can market. I will say that just recently I created a flyer and I put it out in all of the mailboxes in my neighborhood. I think I hit maybe about 100 different mailboxes. And the flyer included details just about, what we were doing from a fundraising purpose. It explained Declan's story, and really went through each of these kind of points. And then it had a QR code on there. So just another example of how easy it is to kind of reach out to your community, get them to help support your event. You can, let's see here. Fundraising boosters are really important. So fundraising boosters are just a way to sort of, you might have kind of your main event idea. That's sort of, bringing you in your money. But then you want to add boosters to it. So like, let's just say, for example, like one of the things that I'm doing coming up soon is I'll be working with a restaurant to host a fundraising event there. And so some of the boosters we talked about doing was bringing in raffles, bringing in kids activities, cornhole, just other ways to kind of reach out to the community, bring people in. That, may not be as closely tied to the fundraising event, but are willing to come out and support and have a good time. And then you're pulling in money through these other sort of fundraising boosters. Okay, you can move on to the next slide. Okay, so peer-to-peer campaign examples. So the first one here that I wanted to cover was the Million Steps Closer. So we know that that's coming up soon, and we usually participate in it every year. Actually, it's become kind of a norm that my cousins will reach out to me usually a month or two before and ask me, are we still doing our walk for the Million Steps Closer? So we usually gather at a local beach and get out, and we all wear our shirts, which you'll see a picture of in a minute. Our matching kind of SDS Alliance shirts, and get out and do a walk. And there's usually sometimes 25, 30, depending on, who's around, right? But the idea is just sort of to kind of get your family friends out there, And then another event that we did for the SDS Alliance was we facilitated a hockey game. And this was actually something that was offered by, so my brother and my husband play in a hockey league, and the hockey league is with a local PBA. And so they heard about our story, and they offered to host a charity hockey game, which, we really were able to sort of kind of pull in quite a bit of funds through that event. It was pretty big. And the nice thing was that they didn't want us to really do much for it at all. So the team really kind of took over and took control and pulled together everything for that event. And in addition to the hockey game, we pulled in fundraising boosters like I talked about. So we had 50/50s, we had raffles. So just other ways to sort of pull in additional, ways to kind of, raise money. So if you want to go ahead and move on to the next slide. So this is the picture of all of my family that gathers every year down the shore in Jersey. This is in Asbury Park. What we did here was, I created the fundraising site through the SDS Alliance, shared that out with all of our family and friends through social media. When we were down the shore, we had created all of these flyers. And so we were handing out flyers to folks that were on the boardwalk walking. We handed it to the local community. We had them posted up in their shops that were all around the beach that day just to help. And in the flyer would have a QR code so that folks can come up and take a picture and donate. So just another way to market and make it a little bit easier for folks that, so that they can donate a little bit easier. We try to all, sort of wear our t-shirts that kind of, sort of pulls everyone together and sends, I think, a nice message. We had a lot of people asking us questions during our walk, like what is the cause? What are you doing this for? And so I think it just helps to kind of get more people involved, raise awareness. Okay, you can move on to the next slide. So so this is really just kind of summarizing, kind of what I talked about previously, which is the annual event of kind of bringing our community together to find different ways to fundraise for the SDS Alliance organization. So I think this is kind of a repeat maybe of what I sort of talked about on the previous slide. So I think we can move on to the next one. Okay, and like I said, yeah, so this is the charity hockey game. We were able to raise, I think, it was just about 10,000, maybe somewhere just around there. And for this one, as far as marketing goes, there was some unique things we did for marketing, actually. And I can't take any credit for it because it was really the local PBA that was able to really sort of market, come up with sort of these unique marketing ideas. But in the township, they had this community billboard that was up on this like major highway. And so they were able to post all the details of the event out on this billboard that was on this highway. And so I think that really helped to kind of pull in different families. It also helped that we were natives of the township where this went on. So that really helped. I had grown up there and, was very heavily involved in the community growing up. And I think that's really all I wanted to get to here, was just kind of highlight some of the other ways we can market. And then here's some pictures of the hockey game. My husband is actually on the left there in that first picture with Declan. And then we had, you can see his cousin is actually right to the side of him there. And then we even had brought in Nurse Bev. So I don't know how many of you are familiar with her or know her, but she worked for Dr. Olson out at CHOP in Philadelphia, and was sort of the guru around, as a nurse for SDS. So when we were first diagnosed, I guess I worked with her. Maybe she was there for probably about a year, maybe 2 at the most, and then she retired. So we didn't get to have a lot of experience with her, but she was a wonderful nurse who did a lot for the SDS patients and really knew a lot about it. Okay, you can move on to the next slide. No, can I finish? 1 minute. So making connections. To me, I guess the best way that I've found over making connections is looking at my family and friends a little bit differently. So over time and over the years, I now look at folks as how can we collaborate and work together in different ways to fundraise. And it's really honestly opened up my door, because if I look at every fundraising event that I've done over the years, it's been through a family member or a friend, or something unique or different that they and how they kind of and their role in the community. So I think looking at your family and friends is a way to have them support and collaborate and look at different things that they're doing, how what businesses are they in, are there opportunities there that you can leverage. It really has kind of opened the door. My father-in-law is really active at a restaurant and is there quite a bit and has a whole group of friends there. And so, we recently just started to organize a fundraising event with the restaurant. So it's really easy once you start to kind of just take a few minutes to think it through and come up with ideas and brainstorm. I think it really does come together. I've also looked at some of the things that our community is doing for events and have sort of piggybacked off of that, or sort of leveraged it, or looked at, okay, how can I do that? So that's another way that I've also have found different opportunities. Okay, you can move on to the next slide. So overcoming barriers, yeah, this would, I would say, is kind of one of the things that initially I feel like I was, in the beginning, it was hard for me. I think one, because I'm not somebody who often sort of tells my story or gets really deep into my story. And I think that was something I had to change to be able to really, get our message out there. So, really just, being as authentic as you can and specific in kind of how the disease impacts your life and your child's life. I think that really does help to kind of get through to people. I'm also not somebody who likes to ask for help. So that was another big thing to overcome, especially with fundraising, right? Because that's really what you're doing, is you're going out and you're asking for help. But I guess the way that I've looked at it over time is, every single person has a different reason or something that's kind of driving them to do things like this, right? So to me, it's for the greater good, right? It's going to really open the doors and hopefully find treatments and a cure. And so we, that kind of mindset has really changed my way of sort of going out there and asking for help. Small steps count. So sharing a post, like I said, that's really a simple way to just get your feet wet and get out there. I also thought the same thing, that I don't have a big network when I first started to fundraise. And I thought, geez, I'm not going to be able to really do any kind of big events. But I was completely wrong, and over time, I think I've pulled in maybe somewhere close to about 20,000 over the past few years through all the different events that we've done. And like I said, I really just worked with and leveraged all of my friends and family to help really make that happen. And so it didn't really need a big network. It was just really, working with some of my family members on how to kind of make this happen. So you can move on to the next slide. So that's essentially sort of my my story and how I've been fundraising over the past few years. And I feel like each year I kind of get a little bit bigger with my events. So if anyone out there is ever having any questions or concerns, or you're just, challenged with how to get started or how to come up with an idea or anything at all, I'm happy to talk to anybody and brainstorm, and then that might open some doors and opportunities for myself and future events. The other thing I will say, that actually Eszter showed me, I don't know, maybe a few years ago, but Canva is an amazing app that can help you. Really, it does all the work for you, but that's how I make all of my flyers now. And it's a really simple app. Once you make your flyer, you can save it and then you can build on it. And it's a nice, and it looks nice. It's visually very appealing. So I think that helps to entice people as well. So thank you. That's our story.
Thank you. Thank you, Rebecca. Yeah, Canva, it's just canva.com if anyone else is interested. And you can use it, anybody can use it for free, but we can also collaborate, like give you templates and so on to help you create the communication materials that you need. Yeah, so thank you. Thank you so much. Are there any questions for Rebecca specifically? And I just want to say it has been so beautiful to watch your family really get more and more impact over the years. So it was just beautiful to watch. And of course, Declan thriving is the icing on the cake. So I don't think there are specific questions for you, Rebecca. So I know you might be pulled into all the directions with the family around, but we really appreciate your firsthand experience and sharing what it feels like and what it looks like to fundraise as an SDS family.
No, thank you very much.
Mm-hmm. All right, so in the interest of time, so we're gonna rearrange the events a little bit. So I'm gonna just give you a quick update about Project Pacer, so the community knows where we are with that and what's coming. And then we might just skip the documentary film altogether, and I can share the link to the film with you at a later time, or if you tell me in the chat that that's what you came here for, to watch the film, we can absolutely still play it, probably starting at 3. So let me know in the chat what you think, whether you want to see the film or not. If not, then after my update of Project Pacer, we can just go into a coffee chat where we can all discuss what we learned and what questions we may have a debrief, and bond. So I'm going into Project Pacer next. Basically, I refer to this as Operation Patient Voice. So all our activities that are centered around collecting and communicating what the patient voice is of an SDS or of the SDS community. So this started off with our PFDD meeting, right? That was an opportunity to share with the FDA and researchers what it's like to live with SDS, what impacts our patients and our families' quality of life, and what would constitute meaningful change. And so this PFDD meeting was recorded and is now available on our website for any drug developer, FDA personnel, or family to watch and get a deeper understanding of what the community is about. And that served as kind of the input to create a voice of the patient report. So that is a specific way of capturing the content of the PFDD meeting in a written format. And we will have the first draft of that ready to discuss with the FDA. And once it's finalized, it will be filed by the FDA, again, for drug developers, researchers, and FDA staff to have an easy access and be able to learn what matters to us. And both of these activities, the PFDD meeting and the Voice of the Patient report, are part of Project PACER. So Project PACER is our larger effort to build the patient voice into the research field. Project PACER is a project funded by the Patient-Centered Outcomes Research Institute, or PCORI for short, and we received a Eugene Washington PCORI Engagement Award. And so this helped fund the PFDD meeting, helped us fund all the travel expenses of the speakers and patient attendees at the meeting, and is also helping fund the writing and creation of the report. And it is also helping fund another 1.5 years of work that's coming up, which includes 2 courses. So a course for patients to really systematically learn about SDS, the science, and more importantly, how to bring the lived experience to benefit the research process in all stages of research. So rather than researchers just asking for us to participate in research when it's all already planned out, we envision us to be able to partner with researchers at an earlier stage so that we can provide input on how the research might be more feasible for us to participate in, or the questions be more meaningful to our life. So that this will be a course for patients to become more effective partners in research. And we'll probably have even a certificate that can come with it when you complete it. And then we'll also have a second course, another course that will be geared towards researchers, because researchers are really amazing at the science, but some researchers have limited experience with how to effectively engage with patients. And so this course is designed to help researchers understand how to leverage our lived experience to make their research more impactful, and also more have them be more successful in applying for grants. It could be any grant, but in particular, PCORI grants are very, very focused on ensuring that the patient voice is represented at all stages of research, and specifically research that is called CIR, or it's called Clinical Comparative Effectiveness Research. So that is what they're mostly focused on. And so we want to help the researchers get more familiar with those concepts so that they can be competitive and successful in applying for grants. And these are not just any grants. And the most recent round of grants that are geared towards rare diseases and cancer are up to $12 million per grant. So I would think that it will be absolutely worth the effort of our researcher community to learn about this opportunity. So originally I was planning to replay Dr. Merker's presentation where she summarized the voice of the PFDD meeting at the end of the PFDD meeting in Cincinnati. But in the interest of time, I'm gonna skip that and send you guys the link to that later. And just say that I'm gonna send you all the resources, links, things we talked about. And there's one more thing I wanted to just call out, which is the Global SDS Action Week and Awareness Day. I personally am not a big fan of an awareness day because I don't see the impact as clearly as when I communicate specific actions that we could take during a week. So on our website, there's a section, SDS Awareness Week, and we talk about, we were suggesting things that we could do in that week that would have an actual impact on research, which would be, participating in SDS GPS, updating your records in other registries you might be participating in, maybe participating in the Million Steps Closer to a Cure fundraiser, or participating in a peer support group. And we also have the option to record a quick video about what matters to you in SDS. There is a tool on that website that you can click, and it will automatically help you create a video. It's a very cool toy, so I encourage you to check that out. So that will be a tool for the SDS Awareness Day / Action Week. So with that, given that we ran out of time, just gonna check the chat if anybody would like to see the film now, and if not, we can just jump into a discussion now. All right. All right. All right, somebody would like to connect with Rebecca, and I'll be happy to connect you guys offline, definitely. Jackie's asking for a little break. So nobody was asking to watch the film right now, so we are gonna skip it and launch right into the coffee chat, which is literally just for us to connect and answer any questions and provides peer support. And of course, if anybody needs to take a bio break or anything, we'll be right there waiting for you.